challenging game. That's why I gave it to you guys, because you'll do
Yeah.
it one way and someone changes one thing and then it's all crazy.
Well, it's just so frustrating because you're blocking yourself half the time, and there's a lot of strategy to
You
it.
got to really follow the planet. You got to really follow the sun.
Yep.
I've contaminated Dr. Sperry with my. I found some really. There's another one called Planet that you would like. Same Game Makers. It's called Planet. And if you're nice, maybe for Christmas. Just kidding. No. And that one there, I think that one's even more fun, but it's not as much. It's more biosphere based and little animals and stuff. Kids enjoy them. I mean, the kids, because they can kind of get into it. They can touch something. But yeah, it's challenging. Okay, your mic and your thing are right there.
Okay, excellent. Now we just need a class.
They'll show up in 10 minutes.
So while we're waiting, do you have any questions?
Will you go over
Do
Ploy?
you want to go over, or do you want? I'll look at your what you sent me right now.
Do you want to do it? We can do it today if you want. I mean,
What do you got to do?
I did some drawings of epithelial cell with the different structural.
Well, crack them up. Let's go, Cal. I want to see them. Come on. I got.
Did you send it through the inbox or through the
yeah,
other
through
one?
Canvas.
Train me up. No, we weren't as studious over the break as we should have been, but we definitely were spending some time on it. So.
Wait, is the final in here or is it in the
It's
testing
in
center?
here
On Tuesday?
on Tuesday.
Okay.
Is there a time after class I can talk to you about my grade?
Yes. So we're going to be talking about all this stuff today,
Oh, okay.
and so
Would it be better if we did it after?
yeah, I'm thinking on Thursday would be good.
Okay. Yeah.
But
let's do
I'm
that.
printing it right now, so we have a copy, and maybe we'll
Okay.
use it today.
And
So
I printed out like a little paper that just has like the numbers because I labeled like all different pieces with numbers and I printed out
yeah,
pages for people to write on it. That was awesome.
I'm going to go put this in.
That's why I got them stinking projects done as soon as I could. Because I was like, I didn't want to fart around that near the end. You know what I mean? It's the last thing I wanted to do near the end.
Yeah.
So I was like, I have to make these done. So
Yeah. I don't know. In a way, I'm kind of glad I waited for this one because it gave me a chance to like draw something.
no, that's cool. I'm just saying for me,
Yeah.
I'm just
no.
one of those I'm not one of those last minute people. I can't do that.
It used to be more like that in high school.
Just don't do that well. I mean, I can do it. I just don't like doing it.
Yeah.
So after today's lecture, I'll think I want to add a little more details to it.
Okay.
But I love the records. This is cool. So these are correct. What you don't have on here is the definitions.
Oh, I didn't know if we were listening.
And there will also be, but we haven't learned about it.
Okay.
And so, and then we'll also have active restrictions,
Okay.
which is another lateral one, so it's three lateral. Basically, that's why I want to emphasize no one comes to the hard
Okay. No, I said till 4.
And then we've got.
Trying to get up to like, oh.
And then down there, we've got, we've got my sister,
Yeah, I didn't sleep well last night either. So.
And then what I would like is if you draw this out and just a little bit more. So you show the picture and show your protein is an expression. And you've already shown it to hash and the activation. And then with
So how are you? Yeah, I started sending emails with those YouTube clips of these lectures, so you guys can get on there. No, they work. They work. I don't know one I don't have with one. I just started changing the settings to public, just so there wouldn't be any restrictions on them. And just because you guys don't know what to be looking for them, so I said starts resending links, so you should be able to see them. I don't know if that helps you at this point, but like for me, I just put them on at work, and even if I don't actually see it, I just put them on to listen to her. I should do that, and it's on YouTube, so it's readily available. You're not digging around looking for stupid files, and so. When I'm getting better at editing, it's just the garbage that we need. I still get little bits of crap, but the sound is awesome now. I got the sound all worked out. Yeah. I know where to put the camera for her to see her handwriting.
So
Just
just
put her on this side or behind this way, her body's in the way of it. So that's her putting it up there, because her other class I did the same thing. I don't like putting it over because everyone's walking by it, but I want to see your handwriting. I can't do that.
Exactly. Because it gets confusing. That is awesome. Okay, good morning,
Stop on before
everybody.
you get started.
Oh,
So
yes.
follow you so we can
Thank
see your
you.
handwriting.
Did I turn that on? It looks like it's on.
Is the lab on Thursday?
Yes.
That's the last day of class, man.
I know, it's crazy.
Think about it.
How many classes are you guys taking?
Just take a few, just 8 hours.
I wasn't statistics.
I just couldn't take. I was gonna take 12, and I just couldn't do it.
Statistics class, you wanted
Okay, whose tests do I have here? I've got Chelsea and Angela. If they come, remind me to give them their tests.
Angela should be here.
Good. Oops, that's not what I wanted. Got some good markers.
All right, speak to the devil. Literally just said your name. Literally.
Okay. One more key point we want to cover from chapter 19, which was the meiosis chapter. If you're going through the PowerPoints, there's a ton of information in 19 that we are not doing. I have some slides to help remind you of basic Mendelian genetics, what's the law of independent assortment, the law of segregation, all that kind of stuff. It's just 1610 review stuff. I'm not going to cover that stuff in class. It's just there if you want to review any of those ideas. No, because the final's a little different this year. Normally it has been in the past, but it isn't now. So I expect you to know some genetics. So that's all there if you need to review. But I'm just going to give you the new genetic stuff, and that's what I'll have on the test, not the basic genetic stuff. All right? So here's the first thing I want you to think about. And before we go over it, we need to just draw a quick diagram. In cell signaling, cells need to be able to talk to each other. The way they do that is you have one cell that is the target cell. It's called that because it is the cell that's going to receive the signal. You have another cell, which could be nearby, maybe 100 cells away, or it could be clear on the other side of the body. All right? This is called, I don't even know what it's called, the secreting cell. This is the one that's sending the signal. So this one packages up signal in vesicles. The vesicles fuse with the membrane, and the signal ends up outside the cell. Does that make sense? All right, so this one's sending a message. The signal is the message. How is this cell going to get the message? It has a receptor, and most receptors are in the membrane. You can also have receptors in the cytosol. So that depends on the nature of the ligand. Most ligands are hydrophilic. They are soluble in the extracellular fluids. because they are hydrophilic, can they cross the membranes? Can hydrophilic things cross membranes easily? Yeah, not very easily. And so most signals have receptors that are in the membrane because the signal can't cross the membrane. The key thing about a membrane receptor is it is a transmembrane protein. So it has part of it poking to the outside of the cell where the signal can bind to it. But what we want to do is have something happen inside the cell. So the whole point of a receptor is when the ligand binds to it, that causes the protein, the receptor, to change shape. Where? Inside the cell or outside the cell? Now, it might be outside, but where do we care about it changing shape?
Are we interested in the cascade inside?
Exactly. We want something to happen inside the cell, so that's where the important shape change happens. So, how do cells talk to each other? One cell secretes a signal. That's the message. The other cell gets the signal by the message binding to a receptor. Then what happens? The receptor changes shape. And if it's a transmembrane receptor, the key place where it's changing shape is where? On the inside of the cell. Bless you. When it changes shape, now molecules that it wasn't interacting with before, it will now interact with because you have the right shape. So now this one interacts with it. That changes its shape. So another one interacts with it, which changes this one's shape. So now it's on and something else interacts. And that turns something else on. Okay, and so you get a series of molecules turned on inside the cell. That is the transduction cascade. So this is your reception step, your transduction step. Most signaling pathways have multiple steps here, but you don't have to. It's possible to activate a receptor and immediately get your response, but usually you have multiple steps. And then you have the last step is response, where you get this cell to do whatever it is this cell was telling it to do. So how can cells respond? They can respond by dividing, in which case, what would the signal be? What would you call the signal? A mitogen would be a signal that results in the target cell dividing. Another thing a cell could do is it could change its shape. Maybe it was round and squishy before, and now it's going to become long and skinny like a nerve cell. Or it's getting a message to differentiate and change its shape. How do you change shape? Think about what determines the shape of a cell.
Aren't your intermediate filaments lining the inside of the cell membrane?
Yeah, so your cytoskeleton determines your shape. So how is a signal going to cause my cell to change shape? It's going to cause me to rebuild my cytoskeleton in some new way. I'll be activating some G protein, Rho or RAC, that will rearrange my actin structure or different things like that. So different signals like that. Okay, you had a question.
I was just saying, but we learned in the last exam, we learned that, like how a neuron fires. You receive a signal, it causes certain things to happen, which causes the signal to move down the axon, arrives at the axon terminal, and releases the calcium or whatever.
So if this was a nerve cell, then the signal would be a neurotransmitter. This would be a ligand-gated sodium channel. And the response, so I wouldn't activate a transduction cascade, I would just let sodium come in. That would change my voltage, which would then activate another thing. And so signaling pathways can get very difficult, get very different, but they all have these basic steps. You receive a signal, you do something to figure out what does the signal mean, and then you get the thing to happen in the end, the response. For this unit, we're focused pretty much on cytoskeleton and cell division. And so I want you to focus on receiving signals where the response is cell division. So now that gets to this question, talking a little bit about genetics and that kind of thing. We learned about alleles and how you segregate the DNA to different things. Would a mutation in a receptor that causes it to act as if a ligand is bound be a dominant or a recessive mutation? And how do you know? Do you remember our definitions of a dominant mutation or a dominant allele means what? Remember the widow's peak with Romney and Ryan? If you have a dominant allele, how many copies of that allele do you need to show the effect? Just one copy. And what's the definition of a recessive allele? You have to have two copies of that allele to do it. So, think about this. How would you answer this question?
So it's basically our receptor in our cell membrane. Instead of it being closed or not active, this mutation is just causing it to act as if it's receiving a signal all the
Yep.
time.
So how would we figure this out? First thing that you'd want to do is think about your chromosomes, okay? We get how many copies of this allele? 2, because we are diploid organisms, right? So we've got 2 copies, 2 genes for this receptor, and one of them is normal and one of them is mutated, okay? So one's mutated, one's normal. What we want to know is the mutated allele a dominant allele or a recessive allele? So what's the receptor doing? It's acting as if a ligand is bound. So let's say that the normal gene is making a receptor that is normal. Is normal receptors on or off when there's no ligand around? They are off. They're only on when there's ligand around. This one's making mutated receptor. And how is it mutated? In a way that it thinks ligand is bound to it, even when there isn't ligand bound to it. So is it on or off, even if there's no ligand around? It's on. So now I've got a cell. One gene is making normal receptors. One gene is making receptors that think they're always on. Is this cell normal or abnormal with just one mutated allele? It's abnormal. Why? Exactly. This must be a dominant allele because this protein will always be on whether there's ligand around or not. And if it's on, will these things be on? There's nothing wrong with any of these molecules, but are they now going to be on when they're not supposed to be? Because I have a mutated receptor. And how many copies, bad copies, do I need to show this effect? I only need 1 copy, so this would be a dominant allele. Does that make sense?
So that is the definition. When we have one mutation, it acts as dominant because there only needs to be one to make it happen.
Exactly.
If it was a recessive allele, then it would need both of them to be mutated.
Exactly. So here's another question. Think about that one. Talk to each other. So in this case, we don't have this mutation. We have one normal allele, we have one mutant allele. And in this case, my receptor has a shape like this. Even if there's ligand around, can it bind to this? No. But if there's ligand around, can it still bind to the normal one? So now, same thing. If there's no ligand around, will this cell be normal or abnormal? It'll be normal because with no ligand, I'm not doing anything, and that's normal. With ligand around, will it also be normal? Because this will now respond and I'll be doing something, or basically this allele's having no effect. To have the effect of this one, how many copies of that bad allele would I have to have? They'd have to both be mutated, so I'm only making receptors that don't work. And now, will this cell be on all the time, or will it never be on? It'll never be on. Okay? What kind of a mutation is this?
Recessive.
This is a recessive mutation. So this is the key thing I want you to learn. In a simplistic sense, the easiest definition of what it means to be a dominant allele or a recessive allele is that dominant alleles do something. Sometimes they do something good, then it's a good dominant allele. Sometimes they do something bad, like this guy, then it's a bad dominant allele. But you only need 1 copy of a dominant allele because it's doing something. Even if the other allele is not doing something, which is what it's supposed to be doing, you're going to be doing something. Okay? Recessive alleles typically fail to do something. You have a protein that folds up so it doesn't go interact with what it's supposed to or doesn't do what it wants. And you don't see the effect of that unless you have both alleles do that. Or in other words, dominant alleles, dominant mutations are gain of function. You do something. Recessive alleles are loss of function. All right? Now, why are we interested in this? Because we want to talk about cancer. All right, so what is cancer? It is a disease of uncontrolled cell growth. And you remember, in the cell cycle, what are the different stages? Okay, what goes here? G1, What's all of this? Interphase. And what's this? M. And what are the phases of M phase? Prophase, prometa, then meta, ana, telo, and then cytokinesis, okay? These are all stages of mitosis. These plus this is what happens in M phase, okay? All right. So in cancer, what's happening is we have unrestrained cell growth, meaning, where are the 3 checkpoints that we have?
G1.
We have a checkpoint at the end of G1 and the end of G2 and in the middle of M. What regulates this checkpoint? To pass through here, I'm asking, will I ever divide? How does the cell answer that question? It's a cell signaling thing. Did I ever receive a signal telling me to divide? If I do receive a signal telling me to divide, I'll turn on molecules that will eventually result in me doing what? My response would be to make cyclin. Which kind of cyclin?
It's a SCK.
Yeah, the cyclin that turns on the CDK that regulates this checkpoint. So this is the G1S checkpoint. So what kind of CDK am I making? If we keep it really simple, it'd be S-cyclin, which binds to S-CDK. Okay, there's actually 2 different cyclins that get made here and 2 different CDKs. There's a G1S CDK also that gets turned on. That's details. But I want you to know there's a CDK cyclin complex that gets activated to take us into S. How does it know when it should make this cyclin? It receives a what?
Mitogen signal.
A mitogen signal, and we activate a cell signaling pathway. The result of which is to make what? Cyclin, which activates the CDK, which then turns on things that get me into S phase. All right? At G2, what are we activating? This is the G2M checkpoint, and what has to get turned on?
MCDK.
MPF. And what is MPF?
MCDK and M cyclin.
Yeah, it's MCDK plus M cyclin. All right, so that's what gets turned on if you pass this checkpoint. That takes us into prophase, because what did this MPF do? It phosphorylates lamins, and remember all that other stuff you learned about? Okay, that takes us into prophase, prometaphase. Then at the M phase checkpoint, what am I asking?
Am I lined up?
Who's regulating this?
A and P. Great.
APC, and I'm not going to go over all that right now, but on Thursday when we do our lab, you'll be reviewing all this stuff. The point I want to focus on right now is what would make cells divide too much. If you screw up the cell cycle, it causes cells to divide too much. Which checkpoint is the worst one to screw up that would make cells divide more than they're supposed to? G1, because that's the one where you're asking, will I ever divide? Usually cells get the answer no. And what do? They go into G0. If something happens, so they think they're getting a signal saying divide, then they're going to be dividing. That is almost always the first thing that goes wrong in cancer. Or cancer is a disease of cell division. Do you have cancer just because you're dividing too much?
No.
No. But when you divide, every time you divide, you copy your DNA. So are you at risk of accumulating more mutations if you are dividing too much? And so the first thing that usually goes wrong in getting cancer is a failure of the cell cycle control. There are 2 key groups of proteins that help us maintain our cell cycle and have everything work well there. These 2 groups are broken into tumor suppressor genes and proto-oncogenes. The job of both of these genes is to make proteins that help us go through the cell cycle normally and not divide when we shouldn't be dividing. So if they both have the same job, why do we break them into 2 categories? The reason is because the way they do their job is a little bit different. Tumor suppressor genes, if you mutate them, tend to be recessive mutations, Because tumor suppressors are proteins that are normally on all the time, which means if I mutate it, it's now not on. How many mutations do I have to not see the effect of it anymore? I would need both of them to be mutated, and therefore it's a recessive mutation to not have the ability to do that function anymore. Proto-oncogenes, on the other hand, tend to be dominant mutations, because proto-oncogenes are any of the genes that code for any of the signaling molecules. In a normal cell that's not seeing a signal, is the receptor on or off?
Without the signal present.
Yeah, if there's no signal, it's off. And are all these on or off? They're all off. So they're normally off. They're only on when they're supposed to be. If I mutate one of these things so this protein is on when it's not supposed to be, is this cell now going to think it's receiving signal and be dividing when it's not supposed to be? So anything in your signal transduction pathway, the receptor or the transduction molecules, is usually a proto-oncogene, meaning it's a normal gene regulating the cell cycle, but if you mutate it, it's now an oncogene. What's an oncologist?
The doctor who studies cancer.
Yeah, so what do you think an oncogene is?
It's a cancer producer.
Gene that causes cancer. And why does it cause cancer? If any one of these things is on when it shouldn't be, what am I doing?
You're turning something on that should be off.
More specific. In this pathway, the signal is a mitogen. What am I doing that I shouldn't be? If there's no mitogen around, but one of these molecules is on anyways, am I going to be dividing?
You're going to have control.
And why is that a problem?
You're going to control it. Go out of control.
Everything else is
Does
off.
it just keep dividing?
Because every time you divide, what do you have to do first?
Replicate your DNA.
Replicate your DNA, and then are you in danger of getting other mutations that eventually make your cells abnormal? Normal cells have to be connected to the correct neighbors and the correct extracellular matrix. If I now get mutations because I'm dividing too much that say, you know what, you can live even if you're not connected to the right neighbor. Now can my cell leave the tissue it's supposed to be in and go somewhere else?
Metastasis.
And I no longer have a benign cancer, I now have a malignant cancer. It can go into nearby tissues, but it can't go anywhere yet. Still has to be attached to something. Then I could get another mutation that says, you know what, you don't have to be connected to anybody. You can just travel through the lymph, travel through the blood, go anywhere you want in the body. And now I've got full-blown metastasis. And why is that bad? Because now I've got a cell that's meant to be, say, in the colon, with all the proteins ready to respond to signals that you see when you're in the colon, but my cell isn't in the colon. It's now in the liver. And in the liver, you're seeing other cells, and might that make my cell do bad things that it ought not to be doing? And that can eventually kill you. All right, so our goal is to understand cancer. First thing you need to understand is we've got these two kinds of genes, tumor suppressor genes and proto-oncogenes. We introduced P53 and RB last time. They are both tumor suppressor genes. Why? Are these genes normally on or normally off? Proto-oncogenes are anything in the signal pathway. They are normally off. And I mutate them so they're on, that gives me cancer, and the mutation is called an oncogene. These fall into a different category because these proteins are normally on. So, now let's put a little more detail in our diagram here. I've gotten kind of messy with all my mutations. So let's just do a normal signaling pathway. Here I have a signal. It is a mitogen. It causes cell division. If there's no mitogen around, all these things are off. And how do I get cell division to happen? In my nucleus, I have my DNA. And there's a gene in here for cyclin. Cyclin S. Okay? There is a transcription factor, and it's called E2F. It is a transcription factor, and this transcription factor has to sit down on the DNA to get this gene to be on. Everybody following? But normally in cells, what is bound to the transcription factor? RB. RB. So RB is on when there's no signal, and it keeps TF, or E2F, off. And am I making cyclin? No. And am I dividing? No, which is what I'm supposed to be doing. So how does this prevent us from getting cancer? It prevents us from making cyclin and going into S phase unless we get a signal telling us not to. Okay? So this protein is normally on. Now, if I receive a signal, I activate the signaling cascade. The last thing that gets turned on is a kinase. What do kinases do? Phosphorylate things, and this kinase phosphorylates RB. When RB is phosphorylated, it turns off, and what happens to my transcription factor, E2F?
It becomes available.
It turns on, and do I start making cyclin? And now does my cell start dividing because we just activated SCDK? Okay. So if this gene is for RB over here, is RB a dominant or recessive allele? RB is normally on. So if I mutate one copy, what's going to happen in the cell? Do I still have some RB that's on?
Yes.
So are my cells dividing when there's no signal? No. But if I mutate both copies of RB, now is RB keeping the transcription factor off? And even if there's no signal, is this gene going to be on? And I'm going to be dividing.
So both alleles for RB have to be affected.
Both of them would have to be affected, so it's a recessive. And that's why they get grouped into two categories, because these are things that are normally on to prevent cell division. They're only off when we receive a signal, turning them off. And so to lose their function, you've got to lose both copies. So it's recessive. These things is anything else in the pathway that's normally off, except when there's a signal. And so you only need one copy to be on when you're not supposed to be to see the effect of the bad gene. So those tend to be dominant mutations. That's what we mean by that idea. Okay? All right. So that's how RB works. It keeps this transcription factor off. What did P53 do? Say it loud. Yeah, it's on all the time, and what's it checking? It's checking is your DNA okay? If it finds mismatches, lots of problems, or broken DNA, then it gets it then turns on transcription factors that cause you to make what? P21 and DNA repair enzymes. What does the P21 do?
It acts as a lock for the CPK complex.
So here what's happened is we've received a signal telling us to divide, which means we've inactivated RB, and we've made what? Cyclin, and my cell will now be dividing. But should I divide? My DNA is in bad shape. I shouldn't divide. And so at the same time, RB is getting activated, and it makes P21. And what P21 does, it binds to this complex and turns it off. So basically what's happening is RB, what P53 does is it says, okay, we got a signal to divide, but hold your horses. Don't divide yet. Or we're pausing the cell cycle. It says, sure, I'll let you divide, but we don't want to divide yet. Why?
We've got to make some corrections.
Because the DNA is in bad shape, we've got to fix it first. How do we fix it? We say, first we pause the cell cycle. Who does that? P21. And we also turn on genes for repair enzymes. If you're able to fix the repair enzymes, then you stop. If the repair enzymes are able to fix the DNA, then you stop making P21. That turns this on, and you go ahead and divide, and everything's good. If you're not able to fix the DNA, then what happens? Yeah, that causes P53 to trigger genes that then cause you to apoptose from the inside out, meaning an internal signal tells you to apoptose, not a signal from another cell.
So is it also P53 then that turns P21 off once the DNA is repaired?
Yes, ultimately it is. It's just when the DNA gets repaired, then the signal that's activating P53 and telling it to turn on the transcription factor that makes P21 stops happening. Okay? So it's a negative feedback loop. All right. So those are tumor suppressors. Proto-oncogenes is anything else. Is that two of the same slide? All right. So that's basically just summarizing what we just said. Same thing. And that also is summarizing what we just said. So you can look at these slides if you have any questions and want to review that. But that's an important principle. Now here's one more important principle from chapter 19. We talked the beginning of class about forward and reverse genetic screens. To understand what's going on in cells, you now understand it's all about what kind of proteins are currently being made and what do those proteins do. So we're forever wanting to identify what are the proteins that are in cells and what is the gene that codes for that protein so we can understand the structure of that protein. So the way that you find let me back up. I didn't say that very well. So if I want to understand, how come we understand all this about cell signaling? Because we have done studies like this to identify all the proteins involved in the signaling pathway to get cells to divide. If this was all the proteins, how many genes are involved? Is there a gene for the receptor? And there's a gene for this molecule, and this one, and this one, and this one, and RB, and the transcription factor. All of those are involved in this pathway. You use forward genetic screens to identify what proteins are involved in a process. So what do? You randomly mutagenize the cells. So maybe I take a bunch of normal yeast cells, expose them to X-rays, some kind of radiation, and then what's the process I'm interested in? Cell division. So now I look for cells that can no longer divide. And those are the ones that I'm interested in. I now get a group of cells that can't divide. And now I want to know why can't they divide? Do you think all of the cells, say I got 1,000 cells that can no longer divide, do you think all of them will have a mutation in that protein? No, because we mutated randomly. So some might have it in the receptor, some might have it in this protein, some might have it in any of the other ones. So what we're doing here is we're making what's called a complementation group. All the mutants that have the phenotype we're interested in are a complementation group. And then what we're going to do is mate those ones to each other to figure out how many steps are in the process. So that's what I'm going to teach you. We're not there yet. So what is a forward genetic screen? It's when you randomly mutagenize, you look to see which children, which organisms can no longer do the thing we're interested in. Now we know we've mutated a gene that's important in that process. And so you look just at those. Once you've identified them, now you can do reverse genetic screens. So the first thing you do in forward genetics is apply some kind of mutagen, then look for organisms that have the phenotype you're interested in, and then you can collect the DNA of those organisms and see how is this DNA different from normal DNA, and you can figure out where the mutation was, and then you can identify, okay, where do I have ORFs in this region? And oh, there's 3 ORFs here. Maybe one of these genes is the gene that we mutated. And eventually you can locate where is the actual mutated gene. All of this is considered forward genetics. So what's the end result of forward genetics? You figure out, you identify a gene that's involved in a process. You still don't know necessarily what it does, but you know where it is. You know what its amino acid sequence is, or nucleotide sequence. Reverse genetics is when you now do experiments with this to figure out what does this do? What does this protein do?
And I'm just asking this. This seems really, really random that you would just take 100 mice and just irradiate them and look for the ones that can't. Does that make sense? That just seems really random.
It actually works though. And they can make it a lot simpler. But here's an example. Well, what I want you to know is just the details. If I want to find a gene, I'll answer your question in a minute, but let me get there. So what kind of genetics am I doing if I have no idea what genes are involved and I don't know where the genes are. I just want to locate them. I'm doing what?
That's forward genetics.
Forward genetics. And what's always the first step?
Just to randomly
do something to randomly screw things up. All right? So if I'm an alien, I want to know how cars work, I come and I get bricks and I start throwing bricks at the car. And I break things. And I see what can't the car do anymore. And I go, oh, this is involved in that process. Okay? I knock the door handle off and I figure out, oh, door handles are for opening the door. I crack the windshield and I realize, oh, that's for looking through and not having wind blowing in my face when I'm driving. Okay? And so that's what we're doing in forward genetics. In reverse genetics, once you've located the gene, you can now put that gene into another organism and see what it can do. You can make proteins, lots of proteins of it. You can do pull-down assays and say what interacts with that protein. Anything that you're doing to understand what the gene does once you have it sequenced is called reverse genetics. All right, so here is an example. These little worms, they tend to feed in groups. I think these are, actually I don't remember what kind of worms these are. But anyways, some kind of soil worm. And these guys feed in groups. So they mutated them. They wanted to know, why do they feed in groups? Are there genes regulating this behavior, or is it something else? So they mutated them, and they isolated worms that like to eat alone and don't like to be in a group when they're eating. So now we know that. So now the next question is, I've got all these worms that I know have that process screwed up. The next question is, how many genes are there in the signaling pathway regulating this response? Are all the mutations in the same gene, or do I have mutations in different genes? How are we going to figure that out? This.
Of the worms that don't group together, we're going to isolate their DNA and then look for the different changes from the normals to the...
That is one way you can do it. And now we can do that because it's so easy to sequence DNA, but it used to not be that easy. You could find a general location where you were, but you didn't know exactly where your gene was. And so what you do is a complementation test, and that's what I'm going to teach you about. But what you said also can be done, but it's not what we're going to cover right now. So what is a complementation test? Here is another example. We've had a mutation. We're doing these funky looking chickens here, and these chickens normally are colored. But we had a mutation that makes them be white. We want to know what genes are involved in making these feathers colored. So I have a group of these birds that are all white. The question is, do they all have a mutation in the same gene, or do they have mutation in different genes? So I take a male and a female that both have the mutation. I breed them to each other. This is my parent generation. And the children are colored. What does that tell you? They must be heterozygous.
The white gene must be recessive because the kids would have the white.
Yeah, so for complementation to work, you have to assume your mutations are recessive, that you've screwed something up so it doesn't work, not that you've screwed something up so it's doing something you didn't want. You also can do complementation with dominant alleles, it's just a lot harder. Okay? What do we know right now? These are recessive, this is recessive allele, and both parents are white. So, if the parents are both AA, and that's why they're white, because they're no longer making pigment, then what would my kids be? What kind of gametes can this one make? Yeah, this one can only make A gametes, and this one can only make A gametes. So when I do my Punnett square, what are all my kids going to be? They're all going to be white too. Is that what happened? So what do we immediately know?
The white gene is dominant.
It could be that the white gene is dominant, but anything I give you with complementation, we're assuming the mutation is recessive. So that is not the case.
It has to be more than one gene.
Exactly. It has to be more than one gene.
It can't be one set of alleles.
So now what do we know? We know that one parent is homozygous recessive for gene 1, but the other one didn't get mutated there. It's still normal. And then there's another gene, at least one other gene in the process, where this one is normal, but this one is mutated. And we know it's homozygous recessive in the mutation because I can't do it anymore. It's recessive allele, you have to have 2 copies. Now, what kind of gametes, so this is a dihybrid cross. What kind of gametes can this parent make?
Big A, little A.
Little a can go with big B, or little a can go with little big B, or little a can go with big B, or little a can go with big B. So if they were heterozygous for both traits, we'd have 4 different gametes. But is that the same in every case? So the only gamete this thing can make is little a, big B. And you'd put it down in 4 places, but you don't need to write them all. What about this one? Big A, little b, or big A, little b, or big A, little b, or big A, little b. So the only gamete that one can make is big A, little b. This is my parent generation. What do my kids look like? They're all going to get a big A from this parent, and a little a from this one, a big B from this parent, and a little b from that one. And are they able to make the A protein? Yes, because they have a dominant allele. Are they able to make the B protein?
Yeah.
Or do they have all the proteins needed in the process, and do they end up with colored feathers?
Yeah.
So what does this tell us? So when you get mutants, when you first do your forward genetics, and maybe you get 10 individuals that all are white, then I breed them to each other. And so if I breed one with 9, I get white babies. But if I breed one with 10, I get colored babies. And if I breed, so we'll say one with, let's see, that one was white. One with 2 through 8 and 10, I get colored babies, but one with 9, I get white babies. That means they're in different complementation groups. They have mutations in different genes. Okay, these two had them in the same gene, so they're a complementation group, but one and any of the others are in different complementation groups. So that's what a complementation study is.
How would you write that, like, ratio?
For my example here with 10 in the group, I'm not sure how you would write that. I'd have to think really hard to figure that out. But if I was to ask you, the way I would say it is, we did an experiment, we got 10 white individuals. When we mated them to each other, we ended up with 3 complementation groups, meaning there were 3 times that when I mate them to each other, I get colored babies. That means how many genes are involved in the process? At least 3. If I had 10 that are white, and when I do every possible cross, mating them to each other, I only get 2 complementation groups. That means there's how many genes involved? At least 2 genes. If when I mated them all to each other, I got 5 different complementation groups, 5 different ways I could mate them and still get colored babies, then there'd be at least how many genes in the process? At least 5. So it's the number of complementation groups that determines how many genes are in the process. And we never know the maximum, but you know at least the minimum. There's at least 2 genes in this process if there's 2 complementation groups. What is a complementation group? It's 2 that I can breed to each other, and I get, if I breed them to each other and I get colored babies, then they're not in the same complementation group. If I breed them to each other and I still get mutant babies, then do you know they have mutations in the same gene? And so they're in the same complementation group. So a complementation group is only when I cross them and I get different groups of babies. I cross them and I'm able to get normal babies. That's the complementation groups. Okay, so that's the last idea that you want to do. So complementation groups are really useful in classic genetics. Now we just look at the actual DNA and it can be a little bit quicker. So the question here, these stickleback fish have a mutation so they get a crooked back, and they both look exactly the same. Or if we did forward genetics, they have the same phenotype. We're looking at what genes regulate whether your back is straight or whether your back is crooked. But when I cross these 2 to each other, I get normal babies. So are these 2 fish in the same or different complementation groups? Different complementation groups, and you know there's at least how many genes involved in this process? At least 2. Okay? Here we've got 2 that I crossed to each other, and they had babies that also had bad backs. So what do we know? They're in the same complementation group, or they both had mutations in the same gene. But if I crossed either one of those to this one, would I get normal or bad babies? Normal babies. So we know there's at least 2 genes involved. Okay? Because I've got 2 different complementation groups. Question, or did it get answered? All right? Okay, so here's a question. I think we had this as, I can't remember, have I ever given this to you as an extra credit question? Well, anyways, think about this one. And I'll just tell you what RAS is. Doesn't really matter, but it's kind of, you'll hear about this protein a lot. RAS is a monomeric G protein. And usually when you have a mitogen that's going to cause cell division, the first thing that gets turned on here, that's usually RAS. And then it turns on the next thing and the next thing and the next thing. And RAS is a gene, about 30% of cancers have RAS mutated in them, so it's on when it shouldn't be on.
Want us just to answer it, or?
Yeah, so talk to each other. What do you think? Okay, so here we've got RTK signaling proteins. We didn't go over that, so that's fine if you don't know that. But you do know this is a transmembrane receptor that's receiving a signal. What kind of a signal is this in this case? Did it talk about cell division? I guess it didn't say what the final response is, so you don't know. But at any rate, it's a ligand. We activate this. It activates something which activates RAS. So this is a monomeric G protein. So what would this be? How do you turn on a monomeric G protein? I'm just reviewing a little bit right now. Remember learning about gaps and GEFs? Which one do you need to turn a monomeric G protein on? A GEF. So this would be a GEF, okay? And then when this is on, it turns on the next thing, which eventually causes some response in the cell. Okay, so that was an aside. Don't get confused by that. So now the question is, which of the following conditions would turn off signaling in this line? So what do we know? We have in normal signaling, we turn on this RAS protein, which is downstream of an RTK receptor. You examine a mutant cell line with a constitutively active RAS. What does that mean?
The RAS is always on.
Means it's always on. So is this a dominant or a recessive mutation? Dominant mutation. And so as long as one of them is mutated, are these cells going to be having the response? So now, what could I do to turn off signaling in this line? Add a drug that prevents X from activating RAS? So if I inhibit X, is that going to have any effect? No, because RAS is downstream of where we're at. So we talk about upstream and downstream. What's the flow of information? The ligand goes to the receptor, goes to the first molecule, the second, the third, and eventually the response. Where am I on when I shouldn't be? RAS. Right here. So anything I do in front of that is not going to have any effect. RAS is still going to be on. So what about the second one? Add a drug that increases the affinity of Y for RAS.
It's already on.
Yeah, that's going to turn me even more on than I already was on. Okay, because this is already on, but now it's interacting even more with the Ys. So that's the opposite of what we want. What about C?
That will definitely stop this.
Okay. If I inhibit Y so it can't interact with RAS, now will I turn signaling off, even though RAS is on?
Yes, that will definitely show.
So is everybody following? Okay.
Well, that isn't even what it was. It's saying that RAS is still interacting with Y, but you're saying that Y can no longer interact with the target. So you just cut it off between Y and the target.
Yep.
It's still RAS is still talking to
Yep.
Y, but you're saying you don't talk to the signal.
So the point here is we use complementation assays to figure out how many steps are in a process, and then we do things by looking at a mutant thing and then adding stuff to figure out where is this gene in it. Is it the first step or the second step or the third step? So
Would this be reversed then if we had done our mutations and done our forward already looked at it and found that the RAS is on, then we went back and started looking at this individually, that would be reversed because now we're targeting this one thing.
yes,
This would be
that's exactly right. This would be reverse genetics. Yep. Okay. So what about these two questions? So now talk about just normal signaling. I don't have constitutive RAS. But if I had a loss of function mutation in protein X, so it no longer works, is that a dominant or recessive mutation? Recessive, but we're assuming we're homozygous for it, okay? So it no longer works versus one in protein Y, so it no longer works. Would I have the same phenotype in a normal cell?
You're shutting them both down.
In a normal cell where RAS is normal, would I see the same phenotype? Yes, because in both cases, when I have ligand, I wouldn't get signaling. What about in an abnormal cell? What about in the cell where I have RAS on all the time? If I mutate X so it doesn't work,
It's still going to do it.
will my cell still be dividing? Because RAS is on. But if I mutate Y so it doesn't work, will my cell still be dividing?
Shut down.
Then it will shut down. So now, by doing that study, can I figure out that X comes before RAS and Y comes after RAS? So that's what I'm trying to point out to you. Okay. So that's the end of that. Then we have just a few more touch-up points. Your wild type protein folds up how it's supposed to and does what it's supposed to. A loss of function mutation could be a point mutation where I just screw up one nucleotide and that made an important amino acid get changed. So even though the rest of my protein folded up normally, the part that actually did something is no longer working. That could be a loss of function mutation. Or I could have a truncation. So maybe I ended the protein early. A codon got changed to a stop codon, so I never made this last part that does the job. Or I could have a mutation, so I just don't make the protein at all. I never had a start codon, or I screwed up the promoter, so transcription factors can't bind to it or something. All of those are ways you can get loss of function mutations. Gain of function mutation means I get some mutation so that it's on all the time. It doesn't have to interact with whatever it normally had to turn on. And conditional mutants are when at one temperature they fold up normally, but at another temperature they fail to work.
Which one would happen more often? Would you be looking at the beginning part, the point of mutation, truncation?
Loss of function tends to be more common than gain of function, just because it's easier to screw things up than to get something to happen. Okay. Dominant recessive allele misconceptions. Dominant alleles are better than recessive alleles. True or false?
That's just garbage. They're not either good or bad.
Yeah, it depends on what it does.
Yeah.
Like the allele for polydactyly, where you have 6 fingers, is a dominant allele, but it's not necessarily a good allele. All right? Dominant alleles are more common than recessive alleles.
Yeah, they're more common. Dominant. It's going to be expressed.
How many people do you know with polydactyly?
With the extra fingers?
Yeah.
Does it count for cats?
No, people. It's very common in cats. Okay? You don't know anybody with that. So are dominant alleles usually are they always more common than recessive alleles? Yeah, it depends on what's been selected for. Most people prefer 5 fingers, so that's what most people have. Okay?
Would we say going the other direction, missing the finger, but they weren't born with the finger, means that they're polydactyly, or would we say that's...
Polydactyly means too many.
Yeah, extra.
But I don't know if they would group those errors in there. They probably have some name for finger mutations that's a general name, but I don't know what it is. Okay, now I'm going to introduce a signaling pathway that's going to be important in the next chapter. All right, so you've just learned the basics of how cell signaling works. There's one signaling pathway I want you to know. This is the Wnt signaling pathway. In flies, it's called wingless. It's the same protein. So we have a receptor. You don't have to know the name of it, but it's called the frizzled receptor. And this receptor is off right now because there's no ligand. Wnt is the ligand. Okay? So when there's no ligand, my receptor is off. That means my first transduction molecule is also off. What this thing usually does is when this is on, it inhibits this protein. What's this protein?
Anaphase promoting complex.
Yeah, anaphase promoting complex. But here it's having a totally different effect. Because what is anaphase promoting complex? Do you remember what kind of a molecule it actually is? What does it actually do? How does it get rid of M-cyclin? It ubiquitinates things. It's an E3 ligase. It adds ubiquitin, and whatever it ubiquitinates then does what?
Breaks down.
Gets sent to the proteasome and gets destroyed. Okay? So this is the same protein, but it's active at a different site here. When you learned about APC, we were talking about it getting turned on when? In M phase. Right now, we're talking about it getting turned on here at this checkpoint. Okay? And so, when there's no signal, is APC on or off? It's on. It's on. And when it's on, the thing that it is targeting right now is not securin and it's not M-cyclin, because we're not in that phase right now. What are we targeting? We're targeting a protein called beta-catenin. Beta-catenin has 2 functions in cells. It's involved in cell-cell junctions. That's where most of it usually is. But it also is a cofactor for a transcription factor called TCF. When beta-catenin binds to TCF, it turns on these genes that allow you to divide. When there's no beta-catenin, is my transcription factor on or off?
off.
It's off. So it's sitting here bound to the DNA, but I don't have the right set of transcription factors. I also need beta-catenin here before I actually turn the genes on. So what have you learned? In this pathway, if there's no signal, my receptor is off. My first molecule is off. My next molecule is on, because the job of this one is to turn that one off. It's kind of like the RB pathway. So when this is on, it destroys beta-catenin, so I don't turn on any of my genes.
So it's ubiquitinating beta-catenin.
Yep.
We will be degraded and taken away.
Exactly. Yep. Now, if I do see Wnt signal, what happens? I turn on my receptor, which turns on this protein, which turns off APC. Now, when I say turns off, it doesn't necessarily mean that it gets turned off for everything that it does, but it means it no longer recognizes beta-catenin as one of its targets. Okay? So now it is no longer destroying beta-catenin. Beta-catenin is stable, and what does it do? Binds to TCF, and we turn on genes that do what? Allow us to divide. Okay? So this is another important signaling pathway. This is a signaling pathway that's really important in maintaining stem cells. Stem cells are cells that are able to continue dividing and making more cells. And almost always Wnt is the signal, or one of the signals, that regulates those. So this is one signaling pathway I'll expect you to know on the next test. Is APC a tumor suppressor or a proto-oncogene? Or neither?
Wait, APC?
Yeah. In this example.
Tumor suppressor, because it's going to shut off, right?
In this example, is it normally on or off when there's no signal?
It's normally on.
Normally on. And if I mutate it so that, let's see, if I mutated APC so that it no longer works, would this be a dominant or a recessive mutation? It would be a recessive mutation. And is this a tumor suppressor or a proto-oncogene? Is it involved in regulating cell division? So is it involved in preventing cancer?
Yes.
Which one is it, a tumor suppressor or a proto-oncogene?
Because it takes both recessive.
It's a tumor suppressor. Yep. Okay? All right. So those are some key things that I want you to understand. All right, we're done with this chapter. And we're ready to start our next one. And I think I'm going to give you a break right now. Just take a short break. But this is the glorious week of the semester, the best semester of the year, because they're having food for finals in the library. So if you go down to the library, they have free food. Down. Okay, so now we are finally into chapter 20. And chapter 20 is a fun chapter. We've been learning about tissues all along. We already learned about what tissues? That's what we're going to learn about today. We've learned a little bit about epithelial tissues, and we're going to look at epithelial more deeply today. And what else have we learned about? That you haven't been tested on. In our cytoskeleton chapter, we learned about actin and myosin and how you can make fibers out of myosin motor proteins. And what tissue are actin and myosin important in?
Muscle.
Muscle tissue. What kind of cells is muscle tissue made of? Muscle cells. So muscle tissue is made up of muscle cells.
Is that a twin question?
So what are the 4 tissues in bodies?
Epithelial.
We have epithelial tissue, muscle tissue, nervous tissue, and everything else, which is all grouped together into connective tissue. The first 3 are super easy. In epithelial tissues, what kind of cells do you have in epithelial tissues? Epithelial cells. In muscle tissue, what kind of cells do we have?
Chris, it says.
In muscle tissue, we have muscle cells. In nerve tissue, we have nerve cells or neurons. Okay, so those are all easy. But in connective tissue, it's called connective tissue because it's connecting those other 3 tissues together. And there's lots of different ways to connect the other tissues together. You can connect them by bringing nutrients to them and messages and signals that are carried in the blood, like hormones. So what's one connective tissue? The blood is a connective tissue. You can also physically connect them together by having extracellular matrix fibers that the layer of epithelial cells is attached to. And so that's a kind of connective tissue. It's called loose connective tissue. You also can have dense connective tissue, where your collagen fibers, instead of being randomly arranged, are all parallel and make thick fibers that makes your ligaments and your tendons, all right, that connects muscle to bone and bone to bone. That's another kind of connective tissue. So there are lots of kinds of connective tissues, but just like the other tissues, the connective tissues have cells in them too. And there's different kinds of cells in there. So that's the one main goal of this chapter, is to learn a little bit about connective tissues. The other main goal of this chapter is to learn in a little bit more detail all the junctions that you find in epithelial tissues. So that's our goal for today. To begin with, cells are squishy and tiny. The only thing holding them together is their plasma membrane, which isn't held together by anything except hydrophobic forces. Remember learning all that? So how can I be this amazingly strong and tough person who can bump into things, who can lift 100-pound weights up and throw hay bales and do cool stuff, when I'm made up of cells that are tiny and squishy and aren't made out of anything? How can I make such a strong organism? Because of cytoskeleton on the inside of the cell and what kind of fibers on the outside of the cell. Extracellular matrix, which is most of our connective tissue, is extracellular kind of fibers. All right? So how can we make solid, sturdy organisms? That's what we're going to talk about today. So you've learned before about the levels of organization. We have molecules, and this is going to be on your final. Remember, what are the 4 atoms that living things are mostly made of? Yeah, hunk. And those get put together into what 4 kinds of molecules? Proteins, carbohydrates, nucleic acids, and whatever one we didn't say, lipids, okay? And you guys know a lot more about all of those things, hopefully, than you did at the beginning of the year. And then you put all those together to make cells, membranes, and the stuff inside the cells, et cetera, et cetera. But then we have to connect the cells to each other, and multiple cells of the same type makes what? Tissues, and we've got the 4 kinds of tissues we just talked about. And then if you put all the tissues together in the right organization, you get organs. So every organ in your body has all 4 tissues in it, but how much of each tissue it has determines what kind of organ it is. If my organ is a muscle, it's mostly what kind of tissue? Connective, or muscle tissue. But does it still have a little bit of connective tissue? Do I have to connect that muscle to the bones? And does it still have some nervous tissue? And so every organ has that. And then you put everything together to make your organisms, and then your populations and your communities and so forth. So how do you go from cells to tissues? How are the cells held together? We're going to focus on 6 types of junctions that hold epithelial cells together. Some of these junctions are also found in other cells, but we're going to focus on the epithelial ones. So at the end, you should know 6 of these, and you should know them in some good detail. How do different kinds of cells come about? Yeah, they come from stem cells, which are dividing, and then they have to undergo differentiation processes, meaning they're getting signals that turn on transcription factors that make them make specific genes that turns you into a liver cell or a brain cell or whatever. So you've learned about all that.
When you say it's going to be easy, that's usually the hardest stuff. Those are some details. That's what I was laughing at, sorry. Because I was like, whenever you say it's simple, I'm like, oh God, we're in for it.
You learned about all this when? Was that test 2 materials? We learned how the central dogma works and how differentiation works and how you regulate genes and all that kind of stuff. All right, how does tissue arrangement in organisms occur? How does your body know to put muscles here and to put brain here and everything in different places? So, go ahead. Pardon? We don't have operons in eukaryotic organisms, but you're on the right track. It's because of the signals you're getting saying, what kind of protein should I make? Am I interacting with this extracellular matrix or not? If you're not supposed to interact with this extracellular matrix, the cell will keep crawling and reaching out lamellipodia and kind of testing and saying, is there a protein here that I want to be attached to? Until it finally finds, oh, there's a protein I want to be attached to. This is where I should live. And how does it know what neighbors to connect to? Cell signaling once again. I have receptors or ligands, it could be either one, in my membrane, and they're looking for complementary receptors and ligands on other cells. And if they find them, then you make a junction with that cell, and now you attach to that cell, and that's how you build tissues. So ultimately, that's what's happening. All right. So what are the 4 tissue types? Here we're going to look just at plants very briefly. I'm not going to test you in detail on plant tissues, but there's one thing I will expect you to know. But I want you to understand that plants, just like animals, have tissues. So we've looked at this a lot in our labs. We have epidermal tissue on the outside. Remember when we did our protein localization and we wanted to see which tissues have cells that had this particular protein in it? And you looked at epidermal tissue and mesenchymal tissue and vascular tissue. All right, so we have organization in those. In animals, we also have organization. So typically, in any organ, we tend to have epithelial cells on the outer layer of any organ. It makes the borders. That's the job of epithelial cells, to make borders, also to absorb and secrete. Underlying the epithelial cells, you have to have connective tissue for those cells to attach to. And then you can have usually some muscles involved that say this is the gut to cause peristalsis to happen. So you have some muscle tissue involved. And you also have nerve tissue, which isn't shown in this particular figure. So you have lots of different kinds of connective tissues and some muscle tissue and some more epithelial cells. Here's the main job of each one of these tissues. So epithelial cells is to make boundaries, to secrete, and to absorb. And their protection. These epithelial cells on the outside of my skin are all dead, and it prevents all the bacteria and things that want to attack me and get inside and get all my good nutrients. It gives them this dead desert they have to get through before they can get into me. So it's also protective. So epithelial cells have many functions. What's the job of muscle cells? Just movement. What's the job of nerve tissue? Just sending messages. What's the job of connective tissue? To connect everything else together, and that can be done in multiple ways. All right. Oh, and then this is getting back to what kind of cells are in epithelial tissue.
Cuboid.
Epithelial cells, which could be cuboidal or squamous or all those things. Okay? What kind of cells do we have in connective tissue? Not all of them are this, but most of them are fibroblast cells. That's the most common cells in connective tissue. In muscle tissue, you have what kind of cells? Muscle cells. And here we're back to epithelial. And then also nerves, which isn't in this diagram. Another thing I want to point out here is in all of the tissues except for connective, most of the tissue is what? Cells or something other than cells?
Cells.
Epithelial tissue is pretty much all cells. Muscle tissue is pretty much all cells. But connective tissue is different. You have fibroblast cells, which are cells that can migrate around. Sometimes they'll connect to each other a little bit, but mostly you don't have very many cells. The job of these cells is to secrete proteins, which then assemble into fibers outside the cells, and they also determine how these fibers get laid down. Do they get laid down like this, kind of randomly, which makes kind of a meshwork? Or do they get laid down like this, all parallel, which can make strong structures like tendons and ligaments? Okay? Who's making these fibers? The fibroblast cells. So in connective tissue, you also have cells, but you have very few cells, and you have a lot of extracellular matrix, which is fibers that are produced by the cells, and this is what connects everything else together. Because the epithelial cells are going to have junctions that connect to these fibers. That connects this tissue to this tissue. And these cells will also have junctions that connect to that, and it connects everything together. All right. Here's another important thing. The original cells were single-celled organisms. Eventually, they figured out how to make multicellular organisms. But multicellularity evolved after the common ancestor between plants and animals occurred. How do we know? Because the way you hold your cells together in an animal is totally different than the way you hold all your tissues together in a plant. So multicellularity evolved twice separately in totally different ways.
Because when when we look at some of the fossils of plants, we see the fact that
Sorry, I forgot to put this back in my pocket.
ferns that have they don't have a stem or anything, they just have one thing coming out, and they talked about, we'll just say that multicellularity came multiple times.
Okay. All right. Why do they differ so much? Because the central key defining feature of animals and plants is different. Animals are motile, plants are what? Sedentary or sessile. They don't need to move, so they can hold their tissues together with strong, kind of inflexible things. We need to move, so we have to hold our tissues together in a way that's still flexible and can allow us to move. So what do plants hold themselves together with? Cell walls, which are made out of fibers secreted by the cells. But what kind of fibers are they? Carbohydrates or proteins? Carbohydrates. We also are held together with fibers out here, but they're more flexible, and they're not as thick of layers of fibers as you have in plants. And we don't call it a cell wall, we call it what? Extracellular matrix. And we have special cells to make those fibers, and that's our different kinds of connective tissues. All right. Here's a little bit about plant cell walls. So here we're looking at plant tissues, and here you're seeing the cell walls in between the plants. And in these fibers that are outside, they have stuff that's called pectin. And what pectin does is it prevents the plant from getting mashed if you step on it. It resists against compression, makes you so you don't get squished flat. What cellulose does is it gives you tensile strength. What that means is if I pull on it, it doesn't break. What gives our tissues tensile strength? You guys already know this from learning about desmosomes.
The desmosomes connect to the intermediate fibers,
Yes.
and then that goes cell to cell and membrane to membrane holding us together.
Okay, so if we start drawing a layer of epithelial cells, you remember epithelial cells are unique in that they have an apical side and a basolateral side. What separates the apical membrane from the basolateral membrane? Which kind? Tight junctions, exactly. Okay, and then here's another layer, and another one. I forgot what I was talking about now. Oh yeah, what makes it so I can pull on my skin and it doesn't all pull apart? So these are tight junctions, and they go clear around the whole cell. What are the junctions that make it so I don't pull apart? Desmosomes. And what do desmosomes connect to on the inside of the cell? Yes, intermediate filaments. And then those go through from cell to cell and cell to cell. And that makes a meshwork that makes my cells strong on the inside. Do plants need intermediate filaments?
No.
No, why not? Because they have cell walls, and that makes them strong. And it's the cell walls that have cellulose fibers outside the cells that protect against tensile strength. You can't just go pull a tree apart, right? All right, plants don't have intermediate filaments, so they don't have much tensile strength, but they don't need it because they have the thick cell walls. But if you remove the cell wall, are plant cells pretty weak? And they're under pressure, so they would actually break. Okay. All right, here's a cross section of a leaf, and what actually makes the plant stand up is partly the cell wall, which gives it tensile strength, but mostly it's what?
Osmotic pressure.
The osmotic pressure, the turgor pressure, which we've talked about before. Okay, here's the key thing that I want you to understand that you're likely to see on this test. So here I have my plasma membrane, and the cell is producing these fibers. So it's secreting proteins or actually these are cellulose fibers, but it's secreting sugars, which then get assembled into these big fibers outside the cell. So we have lots of different kinds of fibers. There's hemicellulose, there's cellulose, and then there's pectin fibers. Pectin fibers make it so you're squishy and you don't get squished. They have resilience. and these fibers make it so you can't be pulled and can't be taken out strong. When a cell first divides, it lays down primary cell wall, which is much more flexible, and then as the cell matures and reaches its full size, it lays down more cellulose fibers, which makes thicker, stronger wall. And then you have a middle lamella, which connects the cell wall of this cell to the cell wall of the other cell next to it. That's all details. I'm not going to go into a lot of stuff on that. What I want you to know is, how does the plant make these cellulose fibers? So here is the cool thing. The plant makes these fibers, and it has control over how it lays down the fibers. It can lay down these fibers so most of them are in this direction, in which case, which direction is it easier for the cell to grow? Can it grow side to side or top to bottom easier? Top to bottom, because the fibers are tightly around it. It can't expand that way, but it can expand this way. So if my plant wants to grow taller, it's going to lay down its fibers where? On the top and bottom of the cell, or on the sides of the cell? Sides of the cell, but then it can continue to grow taller. If it wants to put a branch out to the side, then what's it going to do? Yeah, then I'm going to go this way instead. Now I can't grow up and down, but I can grow side to side. So how does the plant have control over this? Here's my plasma membrane. Inside the cell, we have, what are these? Hollow tubes that are part of your cytoskeleton? Microtubules. And the diagram doesn't show it, but attached to, well, it actually does show it. Here in the membrane, we have transmembrane proteins that are called cellulose synthase complex. What do you think they do?
I don't know, maybe. Sorry, its name tells us.
They make cellulose. Exactly. And how do you make cellulose? Cellulose is a carbohydrate, so what do you need?
Sugars.
You need sugars. So we've got sugars here outside the, well, the sugars are inside the cell. What this does is it takes the sugar from inside the cell and it adds it to a chain of sugars that's outside the cell. So it just adds another sugar. And then it takes another sugar from inside and adds it and makes that fiber longer. Now here's the cool thing. These are transmembrane proteins, but they're attached to motor proteins, which are walking along what? Microtubules. And so this cell has control over where it puts its microtubules, and these walk along the microtubules, so it will build the fibers in the same direction that the microtubules are going. And that's how it has control over whether it lays its fibers this way so I grow taller, or this way so I grow side to side.
Dr. Sperry, we can't see it here, but could we figure out the polarity of the microtubules and
Yes.
what motor proteins are using? If it was actually on here, the positives and negatives,
Yep.
we could tell what they were. And,
Yep.
Okay, so they're still using the dynein and the kinesin.
I believe so, but
just can't
that I
see
don't
it.
know for sure, but I think so.
Okay.
Yep. Okay, what's the evidence for this? Here we're looking outside the cell. These are my cellulose fibers. And here's the same cell, and we're looking inside at the underlying microtubules. And can you see they're perfectly lined up with each other? Okay, so that's pretty cool. So what's the take-home message? How do you get three-dimensional shape if you're a plant?
That's how you lay down your...
It's all involved in how you lay down your cell walls, which is controlled by the cellulose synthase complex, which is controlled by where you put your cytoskeleton on the inside of the cell. Okay? All right, now we're going to focus on animal tissue types. We've already introduced the 4 different kinds. Here's the key thing. You can really think of it as just 2 categories. There's connective tissue, and then all the rest. All the rest are highly specialized tissues that have some specific function, contract or send nerve signals or whatever, and they're all made out of just that one kind of cell. Connective tissue is what's holding those specialized things together. Connective tissue has very few cells and lots of extracellular matrix. The other tissues are just the opposite. They're mostly cells with just a little bit of extracellular matrix. Why do they not need a lot of extracellular matrix? Because they attach to the connective tissue. It's making the extracellular matrix. All right. Just like in plant cells, you have fibers outside the cell to give you strength. We also have fibers outside the cell, but they're not carbohydrates, they are proteins. And the main fiber is who? Collagen, yes. Now I also have other fibers out there, but this is the main one, and there are lots of different kinds of collagens. There's like 20 different genes that you have for collagen, and then you can put those together in different ways. So you can make lots of different kinds of collagen, but we're just going to call it collagen. Another important fiber out here is elastin. And we've looked at elastin a little bit before when we were learning about proteins, but elastin are more branched proteins. And so they're kind of springy. They can stretch and they can bounce back. And so if you pull your skin up like this and then let go, it bounces back because of the elastin, pulls it back again. And if you're young, it bounces back really quickly. And if you're old like me, it takes a long time because your elastin breaks down as you get older. All right, we also have proteoglycans in here. And what those are, what do you think they are, just based on their name?
Proteasomes. That was not my question.
Where would proteasomes be? We're talking about stuff that's outside the cell. Would you have proteasomes outside the cell?
I just thought it was
I know, I understand that. But this would mean what? Protein. This would mean what?
Glucose.
Sugar. So do we have, are these combinations of protein and sugar? And what they are going to do is kind of like elastin, they're going to provide cushiness and bounciness, springiness. So we'll get to that in a minute. So all of those are fibers back there. What was your question, Chris?
lotion. People put lotion on their skin all the time.
Yeah.
Does it do anything in regards to the elastin or anything, or are you just essentially adding oil?
Depends on how much you pay for your lotion. Some of them claim to have elastin in them and all that, but I don't think it can really penetrate that far and do anything. It's just oils to keep you more moist, so this layer of dead skin is less likely to peel off and it stays there and looks nice a little longer. But that's not a very good answer. I'm not an expert in lotion. But if I could
Women that are pregnant, like, oh, put lots of lotion on these stretch marks. And I'm like, I didn't think lotion had anything to do with elastin.
I don't know for sure. I'm sure it interacts with it in some ways and moistens things up and loosens things up, but I'm not exactly sure how. But those are great questions. Okay, so there are 6 kinds of connective tissue. You should know this. There's loose connective tissue, fibrous connective tissue, adipose tissue, blood, cartilage, and bone connective tissue. All the connective tissues you should clarify by knowing what kind of cell is in them and what kind of matrix do they have. So the extreme is bone, which is really hard connective tissue. It's made up of cells called osteoblasts and osteoclasts, and then they lay down a matrix of collagen fibers and also a mineral matrix. So calcium carbonate or something like that is out there, and it makes a hard, rigid matrix, and that's why bones are hard and rigid. The other extreme from bones is what? The other extreme is blood, which has what kind of cells?
White and red.
Red blood cells, whose only job is to carry oxygen with hemoglobin in them, and lots of different kinds of white blood cells. And what is the matrix in blood? You don't have lots of fibers of collagen, we just have a liquid matrix, and that is the plasma part of the blood. So these are the 2 extremes. This is solid, this is liquid, everybody else is somewhere in between. These use osteoclasts and osteoblasts. These use all the different kinds of blood cells. Loose connective tissue has what kind of cells? Fibroblasts. And it lays down fibers generally in a random fashion, and you make a meshwork. Fibrous connective tissue, or tight connective tissue, also has fibroblasts, and it lays down the fibers parallel. And what are the 2 tissues we make? If these fibers are connecting a muscle to a bone, it would be a tendon. If it's connecting a bone to a bone, it would be a ligament. So that's where those structures come from.
What will cause them to align? Would it be like an electrical fall through them?
It's the fibroblast cells. They're producing them, and so they have control over how the fiber is built, kind of like what you saw with the plant cells.
I know fibroblasts also respond to stress. They respond to the direction of stress.
Yes.
If they're feeling stress in a certain way, they're going to build up fibers.
They'll build up more fibers in that direction to strengthen.
Based
to strengthen that area.
Tendon or ligament on the side a lot, they'll start to develop fibers controlling for that. So
Yes.
athletes, that's lateral movement, they have stronger ligaments in those directions.
So what have you learned? Two of the connective tissues are loose and fibrous connective tissues. These make some specific tissues. This is found everywhere else. And they have fibroblast cells, and what are the main fibers they're making? Collagen and elastin. We've also learned blood has lots of different kinds of cells with what kind of a matrix? No fibers, but a liquid matrix. Bones have what kind of cells? Bone cells, osteocytes, which is osteoblasts and osteoclasts. And it makes a mineralized matrix. And then the other two tissues are fat, which is made of cells called adipose cells or fat cells. And they actually make very little extracellular matrix, but they're still considered connective tissue because every organ has a layer of fat around it. And it has a protective function. It gives you a kind of a cushioning effect to protect the organ. And then the last one is cartilage. And the cells in there are called chondrocytes, or cartilage cells, and they're the ones that produce a lot of proteoglycans, which makes you be rubbery and absorptive, and so you have a lot of cartilage and things like in between your knee joints and things like that.
So osteoporosis then is horse bone, but when you look at the connective tissues that we're talking about here, we're talking about the fact that the bones aren't producing the connective tissues to pull themselves together and make the bones more dense. Is that what we're saying with osteoporosis?
No.
It's the
Well-
rapid mineralization of the bone, not the actual lay down of the limb.
Okay, yeah, I was thinking of something different. So what was the last thing you said?
Well, because we're talking about connective tissues, and you're talking about these, and I was like, is osteoporosis, is there a lack of these connective tissues that cause the bone to be able to pull together tighter?
Bone itself is a connective tissue, and it is connecting other parts of your body in a way that allows you to move, and it also gives us protective function, so it protects your internal organs. So I'm trying to think if osteoporosis, what osteoporosis does is it just makes it so you don't have as much of the matrix, the calcium phosphate in there. Isn't it just that you get light, weak bones? You don't have as much of the mineralization. Yeah, yeah. So it just makes your bones, it's a defect in the connective tissue function of the bone. Makes them not as strong as they should be. All right, so now we're going to quickly just go through and look at each of these. But I won't. So bone connective tissue is osteocyte cells, osteoblast and osteoclast. So here's the cell. In the middle of each of these whorls, there is a cell. And then it's laying down lots of fibers. What kind? Mostly collagen. And we also have the calcium phosphate out there. Okay? And so this makes a hard mineralized thing. There's two kinds of osteocytes, and they are called osteoblasts, with the B for build. That's what makes new bone, and osteoclasts, which break it down, okay, or clean it out. And every night when you go to sleep, you've got osteoclasts that are breaking down bone, and you've got osteoblasts that are building new bone. And in a healthy body, they're being broken down and built at the same rate. If you have something like arthritis, is it rheumatoid arthritis? I can't remember which one it is. It's the osteoarthritis. Then your osteoclasts are operating more than your osteoblasts. So each night you get little bits of kind of sharp ridges being built on your bone, but you're not breaking down at the same rate that you're building. And so when you wake up in the morning, first thing, if you have a grandma that has this, she's like, oh, it hurts to get up. Because you have to actually literally break those little sharp edges off. But the more you move, the less problem you'll have from this. So if you get up and first thing you do is do some yoga and bend and turn and open and close your hands, your fingers will last a lot longer. But eventually you'll get all these weird things, and sometimes you see old people, where their fingers just in weird shapes and all that. And it's because the balance of the osteoclasts and osteoblasts is different than it should be.
You're just going to die. You know that, right?
Yes, that is true. I will.
What's the purpose of having both these counteract each other?
It's so that you can build the bone. So even though we think of our bones as being something that's always there, they are very malleable. And just like the cytoskeleton of a cell, your bones are getting adjusted all the time based on your use of the bone. And so if I'm constantly playing tennis and going over here and putting a lot of strain on my foot there, then it will break down bone on the inside part of my foot and build more bone on the outside part of my foot. And so I'm shifting based on the use of it.
Calcium, this is also the reservoir for the body's calcium. When it
Yes.
starts calling for it for neural response and muscle response, it's pulling it from here.
Yes, that is also
You're not
true.
drinking milk when you need it, you're getting it from here.
Yep, that's a great point. Okay, loose connective tissue. This is the main thing that I want you to understand, because this is what's going to be underlying tissues like this one. So, how will we draw it? I'm going to draw it just as a bunch of fibers, which are mostly what kind of fibers?
Collagen.
Collagen fibers. And are there also some cells in here? And these cells are the cells that made these fibers. What kind of cells are they? Fibroblast cells. So by the time we're done, you should be able to do a nice drawing like, well, a nicer drawing like this. Okay, so we've got fibroblast cells, there's the cells, and they're typically not connected to each other, but they can temporarily connect. And they're just crawling around, checking out extracellular matrix that's around, and they're looking for signals saying we should go here and build this kind of matrix, and we shouldn't go here, and that kind of thing. All right, so there's a summary of the 4 things. You should know what kind of matrix each one has and what kind of cells each one has. Is it a liquid matrix? Is it a meshwork matrix? Is it a strong fibrous matrix? Is it a squishy gelatinous kind of matrix like the cartilage? All right, now we're going to look a little more in detail at actual collagen. This is very similar to how intermediate filaments form. Remind each other how that happens. Remember the 3 kinds of cytoskeleton are what?
Microtubules, microfilaments.
Microtubules, which are made of alpha and beta tubulin, which come together as dimers, and then the dimers line up to make protofilaments, and then you get how many protofilaments all lined up side by side? 13 around, and that makes a hollow tube, remember? And so the alpha and betas, they have end-to-end connections of alpha to beta, and they also have side-to-side connections to get alpha to alpha to make all those protofilaments all go together. How do you make actin filaments? There's only one kind of monomer. What is it?
What's actin monomer?
Actin monomers. And they bind to each other, and they have 4 binding sites. So they bind to each other, another one binds here, another one binds here, another one binds here. So you get kind of a thin helix. And then you get another thin helix just like that that wraps around the first one. So that makes thin, flexible fibers. How did we make intermediate filaments? There's lots of different kinds of monomers, but they all have the same sort of shape. It's just the ends that are very different from each other. And they all line up very similarly. So basically you have a directional or non-directional monomer. The original intermediate filament subunit, which if we're in the nucleus would be a lamin. If we're out in the cytosol in skin tissue would be a keratin. If we're in nerve tissue, it would be a vimentin. Okay, so there's lots of different names for them, but they all have this structure. It's a linear or globular protein. Linear or filamentous is the right word. And it has an amino N-terminal end and a carboxy terminal end. Okay, but this has a lot of a line of nonpolar amino acids, which causes 2 of these to do what? Twist around each other, but in the same orientation. So am I still directional? And now I've made a dimer. Remember learning all this? Then what happens? 2 dimers line up end to end, but now they go in the opposite direction. So now I have a tetramer. There's 4 individual units. And am I directional anymore? No, because I have 2 N's and 2 C's on this end and 2 N's and 2 C's on this end, so I'm no longer directional. Then what happens to these units of 4? They line up end to end. So another unit like that would line up here, and another one would line up here, and you make big, long fibers. They also can line up side by side and make big, long, thick fibers, and you get how many going side by side? 8 of them for a total of 32 individual ones in this thing. Is it a hollow cable or a solid cable? Solid, is it flexible or rigid?
It's flexible.
It's flexible. It's like a rope made of many strands. What about microtubules? Are they hollow or solid? hollow, rigid, or flexible?
They have a little bit of flexibility.
Fairly, they're a little bit flexible, but they're more rigid than any of the other ones. Okay, what about actin?
That's really flexible.
It's very flexible. Okay, like this, but not nearly as strong as this. Okay, so that's just reminding you what you learned about from your cytoskeleton chapter. I have updated the take-home test, so I know some of you have probably already been working on it. It's essentially the same as it was before, but it just has some cytoskeleton questions added onto the front. And I took 1 or 2 questions out that had to do with genetics or something that I'm not going to ask you about. Okay, but the majority of it is the same. But make sure you download the new copy so that you get that. All right, so how do we make collagen fibers? Once again, we have individual collagen monomers. Once again, they are filamentous, and they have a lot of what? Polar or nonpolar amino acids. Every 7th amino acid, if I'm remembering right, is a nonpolar one, and that causes individual filaments to coil up around each other. But I don't get a coiled coil, I get a triple coiled coil. So 3 of them coil up around each other. And then I have kind of a globular end, amino and carboxy end on them. So that is my collagen molecule. Then these, multiple of these, connect end to end and side by side and make big, thick fibers. And that's what our collagen fibers are out here. Here we're looking at it in cross-section, and here we're looking at it longitudinally. So you can see there's multiple collagen fibers side by side. Here I've cut it in the middle. What is each one of those black dots? It is one of these, which is many of those all wrapped up near each other. Okay, so it's similar to what I just erased there for intermediate filaments, but not exactly the same. I'm going to go over this a little bit more. So when you first make inside your cell, you're reading genes for collagen, you make individual filaments. They are being made into the ER. Why? Where do you want your collagen to end up?
It's got to get out of there.
It's got to get out of the cell because that's where we're going to make big fibers. And do I put it together into big fibers before I secrete it or after I secrete it? Has to be after because the fibers are longer than the cells. Does that make sense? So you read individual genes, you get 3 monomers in the ER, they twist up into that trimer, and they get packaged up into vesicles. Now you'd have thousands of them in this vesicle, not just one, but they don't assemble into big long fibers until I cut the ends off. So these ends are preventing me from getting the end-to-end stuff. I wish I wouldn't have erased that so fast. All right? This gets secreted, and this is called procollagen, just like pro-insulin and all that you read about in the insulin paper. You've got ends on there, and it keeps getting modified until you finally get the final version. So as long as these ends are on here, you won't form filaments. Then what happens? Once I'm secreted, there are enzymes outside the cell that cut the ends off, and now this piece is attracted to another piece, and it binds here and binds here and binds here, and you get big long fibers, profilaments, and then those attach side by side to make an actual collagen fibril, which is what these black lines are representing. All right? These are very strong. They have tons and tons of hydrogen bonds and also disulfide linkages holding the strands together, and that's what makes our tissues very, very strong.
But then the collagen molecule is polar.
Yes. The individual one had an amino end and a carboxy end, but eventually you cut that off, and once you cut it off, I don't know if how you would still have your amino ends and your carboxy ends. I'm pretty sure they are directional, and things can travel along them because I know that cells crawl along collagen fibers, and they probably know which direction they're crawling, but I'm not 100% certain on that. Let's see. So, are there lots of enzymes that are secreted outside the cell that are proteases? What are they doing? Tell each other. There's all kinds of research going on right now to the importance of the extracellular matrix. We used to think, it's just the stuff that the cell's attached to. But diseases and things are caused by what's going on inside the cell. We're understanding now more and more that the extracellular matrix regulates a lot of what happens inside the cell because the junctions that connect a cell to the extracellular matrix are sending messages saying you're in the right place, you're in the wrong place, and they have a lot of effect on what these cells are actually doing. So there's a lot of communication going on. Therefore, is it important that we make the correct kind of extracellular matrix? Yes. And how do you make the correct kind? Well, you have to have good genes for your collagen, so it will twist up normally. You also have to have good genes for your what? Your proteases that cut this end off and this end off. If you're not doing that properly, are you going to get strong or weak fibers? You're going to get weak fibers. And so there's lots of things that can go wrong, is the point that I'm making. There's lots of genes that are involved in making your extracellular matrix that are involved in diseases ranging from arthritis to cancer. It affects how cells travel, how migratory they can be, all kinds of stuff. All right, so here's a disease, scurvy. Read about that.
More oranges.
Yep, this is the disease that sailors used to get when they were on long trips and couldn't get any fresh fruit. Okay, if you don't have vitamin C, you can't make collagen fibers. Why not? What's the role of vitamins in your body?
They're essential proteins.
They are cofactors. What does that mean? Cofactors or coenzymes? They're something that needs to bind to an enzyme to give the enzyme its right shape. If you don't have vitamin C, can the proteases that cut the ends off of collagen cut the ends off of collagen? No. And so what happens? What's the problem with scurvy? Your legs get all crooked and really weak and it's hard to walk. How come? Yeah, because you're not holding your body together properly and it's getting a different shape. And once you get vitamin C again, now do you start making everything correctly again and you can get over this
For the
disease.
record, scurvy is a sailor's disease.
Yes.
Yeah, sailors get it, Marines, Army, Air Force,
Yeah.
they don't get scurvy. It's the sailors who get it. You have to see and they start doing some crazy stuff and they eat their vegetables and fruit and they get scurvy.
Okay, so anyway, so you should understand this. How do you make a collagen fiber? First, what's this? That is a ribosome bound to the rough ER, and I'm currently making a protein. Okay, this is one collagen molecule. It has lots of polar and nonpolar regions, and that causes it to line up with 3 other molecules. And now I have a pre-collagen here that has its ends on either end. There are enzymes that link these 3 ends together, and that's one of the things that keeps me from forming fibers together. Then I get processed in the Golgi, that cuts all these bonds. Then I get released, and out here there are enzymes that cut the black ends off, and now these guys can attach to each other. You also get lots of disulfide linkages, and you end up with these big long fibers with lots of fibrils in between. All right, there are lots and lots of different kinds of collagens. You don't have to know every single kind, but I think humans have 20 different genes for collagen in them. And so you have different kinds of collagen depending on which tissue you're talking about. So in skin, tendon, vascular tissue, it's type 1 collagen. If you take all the protein in your body and look at it, about 50% of all your proteins are actually collagen. It's one of the most important proteins in your body. And about 90% of the collagen in your body is the type 1 collagen because it's in all of these tissues. Okay? Type 2 is what you find in your eyes. Type 3 is what you've got in your arteries, making your blood vessels be strong. Type 4 is in your cell basement membrane. What that is, your basement membrane is a layer of connective tissue that's right underneath here.
Well, that doesn't sound very important.
It's the stuff, I'm not sure, but you know when you're eating meat and sometimes you get this thin layer on the outside of the muscle that you pull off? Yeah, I think that's basement membrane tissue, but I'm not 100% sure because I'm not an anatomist. Okay, so you've got all these different kinds. The point is collagen really important? Yes, and there are many collagen-related diseases. So answer those 2 questions.
So this is dominant recessive again, right? We're talking
Yeah.
about dominant recessive again. bad.
So collagen has a glycine, which is a non-polar amino acid, at every third amino acid site. Why does that matter?
Isn't that what makes it bond?
Yeah, that's what makes the three things coil up around each other to get all those non-polar things next to other non-polar things. Does that make sense? And you start building the trimer where the three coil up around each other at the amino end. So would you expect collagen mutations to be detrimental if only one of the two copies of collagen is defective? I'm not saying whether it's a dominant or a recessive mutation, but I've got two copies, one is screwed up. So now I'm making some normal collagen with glycine every third one, and I'm making some that's not normal.
It just cannot be as resistant. It won't coil up, right?
Yeah, is this going to be a problem? I've got normal protein, but is it going to be interacting with not normal protein? And so I'm going to have lots of screwed up collagen, even though I have some normal protein around. All right, so most collagen mutations are dominant because since they interact with each other, if one of them screwed up, it's messing the other one up too, even though that one is normal. Here's another question, answer that one.
Oh, sorry.
So what if I have a mutation, so I still have one every third one, but I'm missing it here? Is that going to be a better or worse mutation than if I am missing it at the beginning?
It's better to have it at the end.
Yeah, it's better to have it at the end because now I start coiling up from here, and am I still going to coil up pretty well? But here, I'm not going to be able to coil up very well because you have to start coiling here. The rest of that can't coil, and so you're going to have super weak things. This would be a worse mutation than that one. All right? Okay, here are some examples. So mammals, we've got 20 different kinds of collagens. Collagen is the main component of bone, tendon, skin. So these are the key ideas I expect you to know. You don't have to know every kind of collagen and where they are, but these general ideas. Here are some, here is a cross section. I think this is, it's frog tissue. And you can see how it's kind of like plywood. What makes plywood so strong? It's really thin sheets of wood that they've cut off where all the fibers are running in the same direction. And then you lay one sheet down where they're all going this way, and then how do you lay the next sheet down? Yeah, in the exact perpendicular direction, and then the next one this way again. And does that make it really strong? Because then whatever force I've got some fibers that are fighting against that force. Same thing happens here. You lay down a layer where all the fibers are pointing out of the page in towards you, and lots of ones that are perpendicular to you. And that makes the skin really strong, just like plywood. All right. Procollagen proteinases are located where?
They're just outside the ER.
No. When do you cut the ends off procollagen?
Oh, when they need, okay.
After you're outside the cell. So where would these guys be located? Extracellular. They'd be outside the cell. Would these proteinases be made on free or bound ribosomes?
Bound. We're going to get out of the cell.
Exactly. Okay, does that make sense? All right, good. Okay, so if you have defects in the proteases as well as in the collagen, either one can lead to diseases. So these guys that used to go around in the traveling fairs and say, look at the amazing guy with the stretchable skin and that kind of thing, these diseases are usually due because this one happens to be due to the enzyme that cross-links the fibers and holds them together is defective, so you can't form the disulfide linkages. And so it makes your skin extra stretchy because you don't have as much tensile strength as you normally would. This is what Chris was talking about before, it's the fibroblasts that lay down the collagen fibers. And if I take fibroblasts from an embryo and just put them in a petri dish, they will immediately notice, oh, there's another fibroblast over there, and immediately start laying down fibers to connect the two. Their whole job is to say, where are the cells? Let's hook them together.
So going back to the task you mentioned, we were talking about the lack of being able to stretch like that.
Uh-huh.
Like I knew some trombone people who played the brass instruments but didn't do it correctly and started being able to stretch out their cheeks.
Uh-huh.
So could you technically work your body to be able to do something like that if you?
Well, they probably had perfectly normal skin. They just were too lazy to use their muscles to hold their cheeks in when they're blowing. Because when you're blowing, the force is supposed to go through the trombone, but instead they're having it go out in their cheeks, like this. And so, yeah, it's just bad form. You're supposed to be using your muscles to hold your cheeks in and direct the air into the trombone.
It seems like their cheeks were able to get bigger.
It's possible that it got stretched out from that, and maybe they had a collagen defect. Who knows?
Why don't you try it out?
Yeah, do an experiment. Come back after Christmas. Okay, so that is the end of our, I can't remember if it's the end or not. But now I want to talk about one of our junctions. Why are these extracellular fibers so important? Because this is what allows our other cells to attach to the rest of the body. So underlying every tissue, you'll usually have a layer of loose connective tissue. How do I connect this tissue to this tissue? Hemidesmosome is one. Focal adhesions is another. So I'm going to talk about, I can't remember which one I'm going to talk about first.
Focal adhesion, that was the first one on the slide.
Yeah, we're talking about focal adhesions first. So this is a focal adhesion. The transmembrane protein in this junction is called an integrin. And integrins always occur as dimers. So you need two integrin proteins next to each other. On the inside of the cell, there are adapter proteins, which I'm not going to make you learn, but there's tons of information known about these adapter proteins. And, but anyways, on the inside, these adapter proteins connect your transmembrane protein to what? Actin cytoskeleton. So if this is a focal adhesion, then what is this protein?
Integrin.
It is an integrin. And what kind of fiber is it connecting to on the inside of the cell?
What did you show us in the slide?
Actin fibers. Okay, so these are actin. And there's helper proteins to link them, but we're just going to say we're going to the actin. On the outside, I do want you to know the name of the helper protein. On the outside, so what is this fiber here? This black thing is what? Collagen. And my integrin is a transmembrane protein that interacts with a helper protein out here, which we'll draw in blue. And the helper protein is called fibronectin. Now here's the cool thing about this, is integrins have arms that are bendable right here. This protein is very flexible. So when the integrin is active, it's reaching out and it's binding to this part of what? The fibronectin. And what's the fibronectin attached to? The collagen. So that's how we attach to the collagen. But you know that when cells crawl, what do they have to do? Do they have to release their attachments to the extracellular matrix? And so how do I let go? The integrin just lets go of the fibronectin. So now my integrin, which was holding the fibronectin, lets go and folds up. And now my actin fibers can contract, and I move over here, and then what do? I turn my integrins back on, they reach out, there's lots of collagen out here with fibronectin on it, and I just re-anchor to some new ones again. So you can disattach and you can reattach because the integrin proteins are flexible out here. What do you need to know? What's the transmembrane protein in a focal adhesion? An integrin. There's lots of different kinds of integrins, so you have different integrins in a focal adhesion than you do in some other kind of junction. What's the helper protein on the outside? Fibronectin. And it attaches to what? To the collagen fibers. You can remember focal adhesion, the helper is fibronectin. They're both F letters. On the inside, what does the integrin attach to? Actin. And there are helper proteins there, but there's so many of them, and it all depends on the signals the cell's currently getting that we're not going to get into that detail. All right. So that is what a focal adhesion is. You've already learned about focal adhesions. Now you know a little bit more detail about it. Okay, here's a picture of an integrin in its inactive state and in its active state. And when it's active, what does this active site bind to?
Binding site?
A binding site on the fibronectin, which has two binding sites, one that binds to integrins and one that binds to collagen. Okay? And here's some actual pictures. Here's an integrin that is on or off? Off. And here's one that is on. Its arm is extended out and it's grabbing things. Angela?
Does the fibronectin stay with the collagen, so it lets go?
Yeah. I think the fibronectin is just always attached to the collagen, and then the integrins can let go. And then as the cell contracts and moves up a little bit, it just reaches out, and eventually it's going to find another one to attach to again.
So is that also produced by the fibroblasts?
I think so. Yes. Okay. So, integrins are dimers. They have two subunits, an alpha and a beta. You have to have two of them together to be functional. We have 24 different kinds of integrin proteins in humans. This is details. I'm not going to ask you how many are in humans, but just showing you that there's lots of different kinds, and they are heavily involved in cell signaling to give the cells the. This is how cells know if they're connected to the right extracellular matrix, because I'll have a certain kind of integrin, and it can only interact with fibronectins of this kind of extracellular matrix and not the ones on another one, and that kind of thing. Okay, so what do you need to know? Quick quiz. Where are integrins located? Transmembrane. Which part of the membrane?
On the inside.
The apical, basal, or lateral side? They would be on the basal side. So they're involved in what kind of cell junctions? We're going to talk about two kinds. Junctions that connect a cell to a cell are called cell-cell junctions. Junctions that connect a cell to the matrix are called cell matrix junctions, okay? And so, yeah, exactly. And so integrins are part of what kind of junctions? Cell matrix ones, and you're going to learn 2 cell matrix junctions. You're going to learn, I think, 4 cell-cell ones. All right, collagen is where?
It's in the extracellular matrix.
Extracellular matrix. It's outside the cell. How did it get there?
It's laid down by the fibrins, or fibroblasts.
Yeah, laid down by the fibroblasts or whatever kind of extracellular connective tissue you have out there. And it was produced inside those cells. And when did it get turned into fibers? While it was inside the cell or afterwards?
After it left the cell.
Afterwards. And what do you need outside the cell to get it to turn into fibers?
Collagen kinase from
Yeah, proteinases that cut the ends off, and also lots of other helper proteins that do disulfide linkages. And any of those proteins can get screwed up, which are going to give you connective tissue disordered diseases. I had a student in my 1610 class, and when she started the year, she was using those crutches that have kind of arm things.
polio crutches.
Yeah, like that. And she could get around pretty well. She's an awesome lady. And then by the end of the year, though, she was in a wheelchair. And I got to know her pretty well, and she said she had a connective tissue disorder. But the mutation wasn't in the collagen, it was in one of the enzymes that puts the collagen together. And so all her life, she'd been one of these people that's just super flexible, you know, double jointed, and she can bend around and do things, but she could still do everything. Everything was fine. And then something happened in that past year that just triggered things and made this, whatever mutation she has, be much more severe. And now all of a sudden, instead of just being kind of fun and being flexible, now her joints were so flexible they couldn't hold her up. So at first she needed the crutches to help support, and eventually she was in a wheelchair to help support. And she got super interested in learning about proteins and genes, and this was just in 1610. She got so excited about it. And she ended up going on to go to graduate school, and she's studying that disease. And she's found her niche, which is to be a communicator between the research that's going on and the doctors, because this is one of those diseases that a lot of doctors poo-poo and say it's all in your head. And you know, a psychosomatic kind of disorder. And so she's trying to educate the medical community that this is a real disease and that kind of thing. So anyways, interesting story. Okay, so collagen is extracellular. Where's fibronectin?
It's extracellular.
Also extracellular. And the actin is where?
That's inside the cell.
Inside the cell. Okay, so that's what you need to understand to understand junctions, is know what's the transmembrane protein, what do I connect to outside the cell, and how do I do it, and what kind of cytoskeleton do I attach to inside for each of these 6 junctions. All right, next thing to talk about are GAGs. These are called glycosaminoglycans. These are very complicated and they can exist in all kinds of ways, but basically what we're talking about here is this is going to be carbohydrates that are interacting with proteins. One of the ones you may have heard of is hyaluronic acid. Sometimes you see that in supplements that they say to take that will help your joints feel better and that kind of thing. So this is a very simple GAG, and there's much more complex ones too. So what is the purpose of GAGs? These are basically proteoglycans. They're protein sugar complexes. And so you have proteins interacting with sugars, which attract a lot of water, and it makes kind of a gel, kind of like Jell-O, and it makes a cushioning effect. So tissues that have a lot of compression forces on them rather than pulling forces will have a lot of these kinds of molecules in them that protects you from that, gives you a cushioning effect. Where do you need that?
Your organs.
Not so much your organs, because they're not getting squished a lot. You have bones and things to protect your organs. But in your knees would be the major reason. And yeah, I bet it's in your spine too. Yeah. Okay. And what's the tissue called that has a lot of these in it? What kind of connective tissue? It would be the cartilage. Cartilage is for compression. Tendons and ligaments are for tensile strength. Cartilage is for compression strength. And cartilage has a lot of these molecules in it. Okay? So it provides a very hydrated space around the cells. It has multiple functions. It also makes a really finer meshwork. So molecules that are secreted in signaling pathways, if you have a lot of this around, they're not going to travel very far. They're going to stay nearby because it's hard to get through that meshwork. In meshworks that are looser, signaling molecules can travel a lot farther. So lots of functions.
These also play a role in creating like synovial fluid. If they're in cartilage structures that are providing compression, those. Well,
I bet they're part of
I just was more curious.
that fluid, yeah, but the fluid itself, I think, is.
Secreted by these cells. It's
Yeah.
secreted by cartilage structures. I'll look it up.
Yeah, but it's an actual liquid. But I bet that it's still the fibroblast cells that are producing all that stuff.
Secreted by compression and by movement of the joint, and that I would just make sense.
Yeah, I'm not sure of the details of that. That's another great question. That'd be great to find out. Okay, so here's kind of a picture of proteoglycans. And I find these very confusing, so I've kind of color-coded it here. So the blue things are GAGs, the red things are also GAGs, the black is proteins, and the greens are proteins. So we have some long linear proteins, could be collagen, or in this we have red GAGs. This happens to be chondroitin, which is another supplement that people take for bad joints. This one is also a GAG, and that's your hyaluronic acid molecule. And then you've got proteins interacting with that. And this attracts a lot of water, but it also has a lot of consistency to it, makes a gelatinous matrix. Important for compression. Go ahead.
When people take supplements like chondroitin, do they actually work?
That, I am so skeptical of that. Because I just think, how is it, you're eating it, how is it going to get out to your joints? So,
I'm thinking if you injected it to the side.
yeah, if they're injecting it, I think it would help. But if you're just taking a supplement with that stuff in it, I don't see how it would help. But people swear by it. Some people say it really does help. So, yeah, that's what I would think too. So it's a big question that I have not answered. I'm very skeptical of all supplements because I don't think anything can get anywhere and do any good.
Her mom was getting the injections, the stem cell injections in her knees to rebuild her knees. And I was looking at and chondroitin was the suspension that the stuff sits in, the stem cells sit in.
Yeah.
So they were injecting that in there.
So it's probably the chondroitin that was helping more than the stem cells.
I thought the chondroitin was the suspension that
Yeah.
stem cells sit in when they do.
And I'm wondering if this is actually what synovial fluid is all this stuff. So I don't really know. Okay, this is a slide from the other professor, but he just has a different way of talking about all these tissues. So he breaks them up into supporting connective tissue, binding connective tissue, actually hold things together. This one gives you strength, and fibrous connective tissue, which also gives you strength. So supporting would be your blood and that kind of thing. It protects you, it brings you nutrients and all that. I'm not going to use those terms on a test, but it's another way to think about things. All right, so that's the end of connective tissue. Now we're going to look at epithelial tissue. These slides are from a teacher who teaches an anatomy course. I'm not going to test you tons on the anatomy of it. I just want to remind you of things that maybe you already know. Okay? What do you need to know that I expect you to need to know? Epithelial cells always exist in what? Layers. And what is their job? Or they exist in layers, and because of that, they are sided. So I have a side that always faces a space, and a side that connects to the rest of the body. Which one faces the space? Apical. What connects to the body? Basal. And then you have junctions connecting them together, fibers inside and outside. All right. So different epithelial tissues. Blood vessels, what are they facing? A fluid or a... It's either always a fluid or air. So blood vessels are facing what? The apical face is facing the actual blood that's inside the blood vessel. Skin is facing the air. Lungs are facing the air, so on and so forth. What's the job of these tissues? To protect, to absorb, and to secrete. All right, epithelial tissues can be simple, meaning you have just one layer of cells, then connected to the rest of the body, or you can have multiple layers of epithelial cells. So if it's just one layer, it's called simple. If you have multiple layers, it's called stratified. The other word you use to describe the shape of these is the shape of the actual cells. If they're tall and skinny, like rectangles, they're called columnar. If they are short and fat, like squares, they're called cuboidal. So this would be a simple cuboidal layer. This would be a simple columnar layer. This would be a stratified, and you go with the bottom layer shape, and so it looks mostly cuboidal to me, but it's hard to say in that case. All right, and then squamous means that they're flat. So that's the naming system for that. But the important thing is they're all connected to each other. How? With these 6 junctions that we're going to talk about. So here is the epithelial polarity. You already know about tight junctions, which establish apical. The apical separates the apical membrane from the basolateral membrane. This is the basal side. What are the functions of epithelial tissues? Here we're looking at skin, and it provides a barrier of dead cells that any invader has to get through before it can get to your luscious tissues here that have lots of nutrients in them. It also can secrete, absorb, protect, and all that stuff. For secretion, any kind of gland is going to have epithelial cells in it that then secrete stuff into this space. And then you can either have ducted glands or ductless glands.
So what happens with like pimples? Like what is it that's getting like clogged up that causes it?
I believe there is a duct there, but once again, I'm not an anatomist, so I don't know that much about skin. Anybody know the structure of pimples?
2 glands, 2 sweat glands. There's the...
But it's not sweat glands in a pimple, is it? It's something else, isn't it?
Well, there's sebaceous, which is the oil one.
Yeah, that's what it would be. And are those ducted or not ducted?
They're ducted.
Okay, yeah. So it's the duct that's getting clogged then, I would think. Okay? So it's just reminding you of stuff you know, but that's not what I'm going to be testing you on. All right, they can absorb. Now this is important because the example we're going to use of epithelial tissue is the gut tissue. And what's the primary function of the gut? To absorb nutrients. And you already know how it does that because you learned about lots of different kinds of transport proteins that are in the apical membrane. Where would you have a glucose sodium symporter? In the apical membrane or the basal membrane? Apical. Where would you have a glucose uniporter?
Basal.
Basal membrane. Which one's active, which one's passive, all that kind of stuff you've already learned about. So you've already learned they're important for absorption. We're going to learn today how do they form. All right, underlying the epithelial cells, you have what's called a basement membrane. And this basement membrane is part of another junction. So there's another transmembrane junction. In this case, your transmembrane protein is still an integrin, but it's going to attach to a helper protein, and you make tons of this helper protein, and it actually assembles into this layer. And then this helper protein is going to connect to the collagen. And on the inside, these are not going to connect to actin. They're going to connect to, what are the red ones? Intermediate filaments. So this junction is called What are these junctions called? Desmosomes. The transmembrane protein in a desmosome is a cadherin. The transmembrane protein in this junction, which is called a hemidesmosome, is an integrin. So hemidesmosomes are like focal adhesions in that they both use what transmembrane protein? An integrin. But they're like desmosomes because what do they attach to on the inside? Intermediate filaments, and that's why they're called hemidesmosomes. It's kind of like half a desmosome. On the outside, you have a helper protein here, and that protein is called, I think it's called laminin, and it attaches to your collagen fibers. All right, so there's your basement membrane. Underneath there, it makes a sheet of cells. This is called the basal lamina, and the protein that's in it is laminin. And my drawing is really ugly, but we have a great artist in our class, and for her project, she is going to draw a very beautiful picture of this with everything labeled and give it to you next time to use as a study guide. Okay? So these two junctions are very much like each other. Which one connects to actin on the inside? Focal adhesions. Which one connects to intermediate filaments on the inside? Desmosomes. What does that immediately, hemidesmosomes. What does that immediately tell you about the job of this one? Is it involved in movement?
It's an anchor.
It's an anchor. It's involved in strength to make you strong. But does it still have to let go when the cells are moving? But it doesn't make the cell move. It's these guys that make the cell move because the actin filaments can contract. Okay? Both of them connect to collagen on the outside, but they have different helper proteins. What's the helper protein in a focal adhesion? Fibronectin. What's the helper protein here? Laminin. Don't get laminin mixed up with lamins. What are lamins? Lamins are intermediate filaments that form the meshwork inside the nucleus. And remember, you phosphorylate lamins to get the nucleus to break down? This isn't lamins, this is laminin. And lots of laminin makes this basement membrane. And what's the job of the basement membrane? To make you really strongly connected to whatever extracellular matrix you've got nearby. All right, there's 200 different kinds of cells in humans. Almost all of them are some kind of different kind of epithelial cell. And they line all our body surfaces. They were one of the earliest multicellular things to evolve, cells that are connected to each other. Now we're going to look specifically at the gut cells. So here we are looking at epithelial cells, and these epithelial cells have, let's see, they're convoluted here, but that's not really what we want to look at. What we want to change this drawing a little bit. I always confuse myself here. So how do we make the gut lining? The gut lining is made up of epithelial cells which grow in a way that they make microvilli. Okay, so what are these microvilli? There are lots of epithelial cells connected to each other. All right, if I'm drawing this right. The other thing that we have, though, is these guys go down into deep gut crypts. And don't be using that drawing because that's not a very good one. We'll get to a better one here in a minute. So, in your gut, you have epithelial cells. Some of them have the microvilli on the top. These are your absorptive ones. Other ones don't have that. These are called goblet cells. The main job of these cells is to absorb with the proteins we just talked about. The main job of these cells is to secrete. What they're secreting is mucus to keep everything smooth and gliding as it moves through your tract. Does that make sense? But they're all part of the same epithelial layer, the gut lining. And I did this wrong. Just forget this drawing. I don't know why every year I confuse myself on this.
They have structured like that.
You'd think I'd get it right sometime. All right, so now these cells exist in layers. So we'll get back to that in a little bit, but for now, here are all the junctions that you should know. This is a figure out of your book, and then I've added in focal adhesions, which your book doesn't have. The thing I want to tell you, though, is if I ask you to draw a picture of the junctions, I don't want you to draw just this. This picture is sort of okay, but it's not as good as it should be. I want you to label things. So what are our different junctions? What are these that go connect your membranes tight together? Tight junctions. And they go clear around the whole cell. Then we have a connecting junction that attaches to intermediate filaments. So what are these? These are desmosomes. This picture actually is sort of okay. These, because it's showing the transmembrane proteins. What I don't want you to do is just show a little blob like I've got shown here. What's the transmembrane protein in a desmosome? It is a cadherin, and cadherins are proteins that on the outside part of the cell, they attach to another cadherin. So do I need any helper proteins here? They're directly connected to each other. And on the inside, you have a helper protein, which we're not naming, and then that connects to what? Intermediate filaments. The thing I don't like about this picture is they draw the intermediate filaments like a little brush. But do these end, or do they go clear across the cell? They go clear across, and I think it makes a lot more sense to show that they're going clear across, so you see the connections. And intermediate filaments from a desmosome can come down and attach to a hemidesmosome. You're making the whole cell be all connected together. So I just would like you to show those connections. Gap junctions are the other kind that we have, and I'm only going to talk very briefly about them. What they are is they are proteins in this cell, and here, and they end up making a little channel between the two cells. And there's a great variety of proteins they can be made out of. So channels will be specific for certain molecules to move through, not just any molecule to move through. But these are communicating junctions. They let signals that are in here diffuse into here, and I don't have to actually secrete a signal, have it get picked up by a receptor to get a response. I can just have my signaling molecule move directly into the next cell. So what's the function of gap junctions? They are communicating junctions. They let stuff move directly from this cell into this cell. I don't like the way they drew it here. I think it's easier to understand the way we drew it here. You just learned about this one. The last one we need to know is adherens junctions. Adherens junctions are like hemidesmosomes in that they have what kind of transmembrane protein? Cadherins. And cadherins attach to each other on the outside. That's what holds the two cells together. On the inside, they're going to connect to cytoskeleton. What kind? Actin. So, getting back to our drawing, what kind of cells junctions are these? These are our tight junctions, and they're made out of proteins called claudins and occludins, and they go clear around the cell and separate the apical membrane from the basolateral membrane. These are desmosomes, and what's a better way to draw it? If I make my cells not quite so close together,
They have to kind of cross and attach to the desmosome.
yeah, I would have a transmembrane protein. What kind is it? I'll draw it down here a little bit. Those are cadherins. And what do they attach to on the inside of the cell? Intermediate filaments. We've got one more kind of junction. It also uses cadherins as the transmembrane protein, but on the inside of the cell, it's not connected to intermediate filaments. It's connected to what kind of filaments? Actin filaments. So I guess actin we were drawing in black. So it could come down here and attach to a focal adhesion. It could come over here. And, oh, actually, that's not true. I lied. These ones also form a ring that goes around the top of the cell. And so these guys are going all across here, and they're attaching to actin right here. And because it's attached to actin, it can be involved with myosin motor proteins, and you can contract the cell here and make it narrower at the top or make it wider at the top. So what are the two junctions that interact with actin? These ones, which are called adherens junctions. What kind of transmembrane protein do they have? Cadherins. And what do they attach to on the inside of the cell? Actin. And these form a ring around near the top of the cell too. They're in pretty much the same location as the tight junctions. What other junctions connect to actin? Focal adhesions. And then the last, I guess we've got them all. We've got 4 lateral now. Tight junctions, adherens junctions, desmosomes, and gap junctions. Okay? And that's all our junctions. What am I really going to expect you to know? Which kind of junctions have cadherins as their transmembrane proteins? Lateral ones or basal ones? Lateral ones. Which lateral ones? Yes. Which kind of junctions have integrins as their transmembrane protein? Basal ones. And what are the 2 basal junctions? Focal adhesions and hemidesmosomes. Okay? So that's all the junctions. And you should know those. These are 4 cell-cell junctions, 2 cell matrix junctions. Here is a picture of what kind of junction. I've got a transmembrane protein that is an integrin. It's attached to stress fibers made out of F-actin. Right away, you know it's what? I've got an integrin, so it's a basal junction, and it's attached to actin. It must be a focal adhesion. Okay? How does it connect to the collagen fibers? It has a helper protein called fibronectin. What's the other kind of basal junction? Hemidesmosomes. How is it like a focal adhesion? It also uses what? What's the transmembrane protein? Integrins. How is it different from a focal adhesion? What does it connect to on the inside? Intermediate filaments. And here they just said keratin because they're talking about skin cells, but it's some kind of intermediate filament. All right? Do all cells have intermediate filaments? All eukaryotic cells?
No, plant cells don't have them.
All eukaryotic cells have intermediate filaments because all eukaryotic cells have a nucleus. And what lines the nucleus? And what lines the nucleus? Lamins. Do all eukaryotic cells have intermediate filaments in their cytosol? This is what you were thinking of. No, plant cells don't because they don't need it because they have cell walls. But we do. And what do those connect to? On the basal side of the cells, intermediate filaments connect to what? Hemidesmosomes. On the lateral side, they connect to desmosomes. Desmosomes. Not all eukaryotic cells, though, have intermediate filaments. Only eukaryotic cells that are going to be under stress. And so cells that are in layers usually are under some sort of stress. So epithelial cells have them. Most cells in your body don't have intermediate filaments in their cytosol, though. But if I just say, do all eukaryotic cells have intermediate filaments? Then they do, because they've all got it in their nucleus. It's only in the cytosol. Most cells don't have it in their cytosol, but some do.
Like epithelial cell like groupings are kind of similar to like bacteria, like colonial bacteria. Isn't it?
Yes. Yep, it is a lot like that, and that was the original evolutionary thing that happened to eventually develop all these proteins. Okay, both of these basal junctions operate through integrin transmembrane proteins, so that's the key idea we're trying to get at here. So here's a little quiz. We just went over this, so I'm not necessarily going to do it again, but I guess we can really quick. Shout it out. What's the transmembrane protein in focal adhesions and hemidesmosomes? Say this one first. Integrin and integrin. What's the cytoskeletal connection? This one is? Actin. This one is? Intermediate. What's the protein connection to connect to the extracellular matrix? In focal adhesions, which starts with F, we use fibronectin, which starts with F. And in hemidesmosomes, it's laminin. And what does the laminin make? There's so much laminin underlying here that it actually makes a layer, which is called the basement membrane. All right, are there intracellular adapter proteins? What does intracellular mean?
Inside the cell.
Inside the cell. And yes, in both cases, but I'm not making you learn the names of them. All right? And then, is this a cell-cell or a cell matrix junction? Both of them are cell matrix. Okay, and you should be able to answer similar things for the lateral junctions. All right, let's look a little bit more at tight junctions. You already know tight junctions are around the top of the cell, and they hold the membrane tightly together. They are quite complicated in how they form. What I want you to know is what are the proteins that are used to make them? Claudins and occludins. So if you see claudins and occludins, you know they are in the tight junction, and they hold the membrane of this cell tightly to the membrane of that cell. And you would only find them where? In a ring around near the top of the cell. And what's the job of the tight junctions? It makes you so you are not leaky. Even things as tiny as water molecules can't get past this tight junction. What's the other job of them? It separates the apical membrane from the basolateral, so proteins that are here can't diffuse down into here. All right, cadherins. I've told you that we have cadherins as transmembrane proteins in these two junctions. All cadherins work the same, but there's lots of different kinds of cadherins. So in epithelial cells, you have E-cadherins. In neural cells, where nerves need to connect together, you have N-cadherins. Or this is one of the ways that different cells recognize each other. They're called cadherins because they are calcium-dependent adhesion proteins, or adherins. To adhere means to hold together. So as long as there's calcium around, and where do we have high calcium in cells? Outside or inside?
Outside.
Outside. So is there typically calcium out here? As long as there's calcium around, these guys will interact with each other and hold the two cells together. If you want them to stop holding each other together, just remove the calcium and they will break apart. On the inside, there's helper proteins that connect you to what if you're an adherens junction? What would this fiber be? If it's an adherens junction, that's the one just under the tight junctions, and it's actin, and it lets cells contract. If this was a desmosome junction, then what would this fiber be? Then it would be an intermediate filament, okay? All right, so these are calcium dependent, and the same kind of cadherin binds to the other same kind of cadherin. It's one of the ways that our body organizes itself, is when cells are differentiating, they start making either E-cadherins or N-cadherins, and then they know, I should be part of this epithelial layer, or I shouldn't, I should go be something else. Okay. Why did I say not? Oh, yeah, I don't like this diagram just because it's so disconnected from all the rest of the cell. It's better to put them all together. And Cal is going to draw a gorgeous picture for us. Okay, where are the adherens junctions located? They are belt junctions because, like tight junctions, they form a ring clear around the cell, and they're only up here near the apical side. So the only place you'll ever find adherens junctions is around here, but all the way around the cell. Is that true for hemidesmosomes? They can be anywhere below these junctions, and they're just a spot here, a spot here, a spot here, but they don't go clear around the cell. So that's a difference. What's the similarity? They both use what as their transmembrane protein? Cadherins. So you should know how to adherens junctions, AJs, differ from desmosomes, from tight junctions. Here it's totally different proteins. Here it's very similar structure. They both use cadherins, but what they're attached to on the inside is different. One's actin, one's intermediate filaments. So if you just sit and draw that out, sounds like a lot of information, but it isn't that much once you organize it in your head. Okay, here's a movie of some adherens junctions.
Yeah, for the 6 or 7.
These epithelial cells express green fluorescent cadherin. They're grown at low density so that isolated cells can...
Okay, so what we're going to be looking at here is we've attached a green fluorescent protein tag to the cadherin proteins. Where do you expect it to end up? In a cell that's an individual cell, it's not connected to anybody else, where would you expect to see green? It's a transmembrane protein, so it's going to be in the membrane, and it could basically be anywhere, right? But once you start forming junctions, they're going to all end up right on the edges of the cell, and you'll see a lot brighter green there. This is what this movie is going to try to show you.
Initially, labeled cadherin is diffusely distributed over the whole cell surface. As cells crawl around and touch each other, cadherin becomes concentrated as it forms the adhesion junctions that link adjacent cells.
It's alive.
Eventually, as the cell density increases further, the cells become completely surrounded by neighbors.
Here's another question. I kind of stopped at a bad point, but see how this one's kind of reaching out? That would be a lamellipodium. It's pushing outward, looking for another cell to connect to. And eventually it finds it, it forms the connections, and now all the cadherins are just right along the border of those two cells, and you can see it a lot
Or
better.
a tightly packed sheet of epithelial cells.
All right. So here's our adherins proteins. These happen to be E-cadherins. They have multiple repetitive regions, but we don't need to get into all that detail. And they are calcium dependent. That's why they're called cadherins. So what are the two lateral junctions that give us strength? Desmosomes and adherens junctions. Which ones allows the cell to change its shape and move? Adherens junctions. Which one just gives strength? The desmosomes, yeah. Okay, so now here's a key thing. Our bodies are built out of tubes primarily. Well, how do you make tubes? We start out with flat sheets of epithelial cells, but at the top of that, I have a ring of adherens junctions, which is connected to actin. Now, what happens is the cells can change their shape. If I constrict these, it makes my cell narrower on the top and wider on the bottom. If I don't constrict it, but I am constricting in other places, it makes me narrower on the bottom and wider on the top. So I go from having a flat layer of cells, where they're all the same shape, as I start to change the shape of some of them to be like this, what's it doing to my layer? Is it bending it and making it turn? And unfortunately, I drew that the wrong direction. So how do I get it to bend like this? Yeah, I'm shorter here and wider. Let's see, that's not right. Is that right, or am I wider on the top? No, I'm wider on the top and shorter on the bottom. So you make these triangular shaped cells instead of rectangular, and that lets me bend. And you continue doing that, so a flat sheet can go from being a flat sheet to being a little bit bent, to being more bent, to being so bent that those two edges come together. And at this point, there's cell signaling going on, so these cells are being told to make E-cadherin, and these cells are being told to make N-cadherin. And right here at the junction, the ones that are making N-cadherin want to attach to the other ones that are N, and the ones that are making E want to attach to the other ones that are E, and you actually can break this part of the sheet off from that sheet because they're no longer attracted to each other. And I just made what's going to eventually become my skin and what's going to eventually become my neural tube or my spinal cord and my brain, okay? And all of your organs form this way eventually from flat sheets which fold and make tubes, and then the tubes fold more and more and more, and you eventually do that. Is it critical to have adherens junctions to be able to get these shape changes? So that is very important. All right, so here is a picture of a developing embryo, and this is just when you're just starting to pinch off. So here are epithelial cells that are expressing E-cadherin. Here's also E-cadherin. These are expressing N-cadherin, and a little bit later on, these cells have attached to those cells and made a neural tube, and these cells have attached to those cells and made a solid layer over the top of that tube.
Oh, okay, so the first picture, they don't have an epidermis, that's just the inside.
So this is epidermal tissue. Remember when we go through gastrulation and that little ball of cells where they're all the same? Now, the cells that never went through that pore and crawled to the inside of your embryo, they never got the signal to become endoderm, and so they are on the outside and they are ectoderm. And what does ectodermal tissue eventually give rise to? Two fates. You either become the skin or your epidermal tissue, or you become your nervous tissue, your brain and spinal cord. Okay?
I was looking at that, I'm like, oh, there should be this big long scar down all of our back where those two things come together.
Yeah, but there isn't, because this is when it's still just forming, but eventually these cells will attach to those cells and it makes a solid continuous layer, and that will eventually be your skin, and this will become your brain underneath. All right? So early on in development, the first part that you seal is in the middle of your back, and then you close up towards your tail and you close up towards your head. And so the very last thing to close is the very end here and up here by your head. If you fail to close properly here, what disease do you get? Spina bifida. And that's not a good thing, because this is supposed to close over, and if it doesn't, then your spinal fluids, which have a